Research Symposium. Just want to give you a couple just overviews and then we'll proceed. So our main lecturing is going to occur here. We'll have oral presentations between now and 11 a.m. Then we'll have our keynote speaker. That will take us up to lunch. Lunch will be from 12 to 1, and then from 1 to 3 we'll have our poster session. And so we welcome everybody to actively participate throughout the day. And if you go out the doors, if you take a right and an immediate right, there's a set of restrooms there. There's also a set of restrooms going through to the left through the double doors and staying on that main floor. For lunch and for the poster session, if you haven't already been there, it's up on up the stairs above the library in the building adjacent to where we're at. Okay. So with that, I would like to introduce to you the Dean of the Kirksville College of Osteopathic Medicine, Dr. Margaret Wilson.
Thank you. Thank you, Dr. Degenhart. And thank you all for being here today. Welcome for those of you who aren't typically part of our campus. I know we've got lots of students and other faculty from other institutions, particular Truman. We have a wonderful guest speaker that I look forward to a little later in the day. And what a great day to be together to share and to learn. I took a little quick swing around the posters as they're going up this morning, and just so impressed by the work that you all do. So thank you again for being here. We look forward to hosting you here on campus, and have a wonderful day and get an opportunity to meet some new people. So thank you, Dr. Degenhart.
Thank you, Dr. Wilson. And so with that, I'd like to introduce to you the moderator for this morning's program. This is Dr. Jeremy Hauser from the Department of Anatomy.
Thank you. We're going to keep this simple. You guys most of you have been here before. So we'll have about 15 minutes per speaker. If we get done early, we'll have more questions. If we don't, always realize there's the poster session, and you will be able to ask questions there as well. So just to stay on time and to be cognizant of your time and efforts, and to make sure we have enough of a break before the keynote, we will try to stay on time as best we can. So our first speaker is going to be from the Truman State University, right? Kirsten Peterson.
All right, hello everyone. My name is Kristen Peterson, and today I'm going to be talking about disulfide bond-forming enzyme and its expression, purification, and activity. I'm here on behalf of the Dobson Lab over at Truman State, and we have a lot of really interesting research to present to you today. So first I'm going to talk about protein misfolding because it's pretty important in our lab. So what proteins like to do is they like to fold to their most energetically favorable state, their native state, which is where all of their intramolecular interactions are properly aligned. Now sometimes proteins will fold partially, and they'll have certain amino acid side chains that are exposed that can then interact with other proteins, causing intermolecular interactions that will form aggregates or amyloid fibrils. One example of an intermolecular contact is a disulfide bond. So a disulfide bond is a bond between two cysteine amino acids, and it's between specifically their sulfurs. Now, what these intermolecular interactions can sometimes cause is diseases from the aggregates that are formed. So some specific diseases that are formed from disulfide aggregates are prions, ALS, and some forms of Alzheimer's. Now, your body doesn't like this. We don't like diseases. So we have a mechanism to reverse these incorrectly formed disulfides. So an example of a protein that can do this is protein disulfide isomerase. It's a cysteine chaperone, and what it does is it has internal cysteines that will go in, it will interact with the disulfide bond, cause a rearrangement in the structure, and then cause the protein to form back to its native state. Now, the protein that we work with in our lab is disulfide bond-forming enzyme, or DBF. DBF is about 10 times smaller than PDI, and so that means it's about like 7 kilodaltons in size. It comes from the hypothermophilic bacteria Staphylococcus Aureus, and it's pretty awesome. But the thing is even though it has been shown to correct incorrectly formed disulfides, it doesn't have any cysteines in its amino acid sequence. So as you look over here, there aren't any cysteines, and this is pretty crazy to us. How can it do a similar function without the seemingly important amino acid in its sequence? So in order to do any testing on this, we first have to make DBF. So what we do is we streak a plate with the pet vector of DBF, and we grow up a culture, and then once we get it into the large culture, we will induce with IPTG. Then we spin down the cells and lyse the cells and get a protein solution that we can then run on a column. And what that does is our protein will bind. We wash out the other proteins with buffer, and we increase our buffer concentration to knock off our protein. Then we use SDS page to analyze our protein from the fractions that we make. So this is an example of an FPLC chromatogram from a nickel purification of DBF. So what I want you to notice is that this blue line is showing the absorbance at 280 of the fractions, the liquid that's coming off the column. So higher concentration means more things coming off. And then the green line over here is the percent evolution buffer. So we use a gradient and we increase the concentration over our fractions. And then lastly, this red part is where we're collecting fractions. This is where our protein generally will lie, and we collect them in tubes and we can later test them. So what we do, since we focus on this area here, we zoom in and we get a more particular graph. This is showing us even smaller changes in absorbance, and we can see where protein is coming off in the column. So from that, we run a gel that corresponds with our zoomed in version. And so these were the fractions that were taken and run on the gel over here. So what you'll notice is that this first section corresponds to this first bump in the graph. This is where the proteins that we don't want are coming off in the column. So they're not at the spot that we are looking for. So we know that there are other proteins, contaminants is what we call them. And then over here you'll notice this is where DBF comes off. It comes off between 11 and 17 kilodaltons, and that's pretty awesome. So once we've got DBF, then we can start to test it. We do lysozyme assays, and basically we have to use a model protein in order to see if DBF can do certain things. And we used lysozyme because lysozyme has internal cysteines that are all bound up in disulfide bonds. So we can be specifically testing DBF and its activity belonging to the disulfide rearrangement. Now, what we do in the assay is we take lysozyme and we denature it using DTT. So basically, you'll get this like kind of spaghetti soup. It's not going to be a functional protein. It's not correctly folded. And then what we do is we incubate it with DBF and ATP, which will hopefully get our protein, like the lysozyme, back to its native state. Then what we do is we introduce substrate and then monitor the absorbance through that. So this is an example of a graph looking at what the cases are in this experiment. So if you're looking at denatured lysozyme, you should not see a change in absorbance, meaning it's not doing its, performing its activity where it's cleaving the substrate. On the other hand, if we have native protein that is properly folded, you will see in this red curve that it is reducing the absorbance because as it cleaves the substrate, it is absorbing less. So one of the awesome members in my lab, Alicia, has been working a lot with the lysozyme assay. This was our first case. We incubated DBF and ATP and the controls at room temperature for 15 minutes. Now, this is the graph that we got after we normalized. But the blue is no lysozyme, so we're making sure that the substrate's just not sporadically cleaving itself. And then we also have no DBF but ATP and DBF with no ATP. So these are the cases like DBF, since DBF needs ATP, we should be seeing that this isn't actually working. But our test case is adding DBF and ATP, and this is where we should have DBF performing its function to refold lysozyme. Now, what I want you to notice with this graph, this was our first time we did this. You're not seeing an appreciable difference between the changes in absorbance. So our controls are really similar to our test case, so we knew we needed to change something. What we changed first was temperature to see if maybe DBF was working better at a higher temperature, and you'll notice that the changes are not significant again. You're just seeing about the same numbers here, but what we did next was we thought that maybe time was doing something, that maybe if we gave it more time to do its job, it would be able to do so. So you'll notice that we do have a little bit more of an appreciable change here in the changes, and you'll see the green line is DBF. We're hoping to further with this and see if we can get an even better graph with more change between the controls and the test case. So in the future, like I said, we're going to be working with time. That's our first variable that we want to change. We want to see if maybe 45 minutes or an hour is better, or things with DBF or substrate concentration. And then once we have nailed down the lysozyme assay and we know how DBF uses its activity, we can do site-directed mutagenesis and change some of the amino acids to see which ones are particularly involved in its mechanism. Then we have some awesome other members of our lab that work with the gamma crystallins, which are proteins that aggregate with disulfide bonds. So we want to see if DBF can reduce these aggregates and maybe even work to prevent them. And I would like to give special thanks to my research lab. Dr. Cassidy Dobson is amazing. She's been so supportive and super helpful. And also my research group. All of them have contributed so much to all the work that we've done, and we really appreciate that. And also, I'd like to thank the Truman State University Chemistry Department for letting us do our awesome research. Any questions?
All right, we do have time for questions, and when I'd like to ask you to try to share the questions, and maybe one question per. Brian will have a mic, I'll have a mic as well.
Since you're using the pet vector and using a cobalt or nickel, that obviously suggests you've got a his tag on this. Mm-hmm. Is it on the amino or the carboxy terminus?
It is on the N terminus, and we use a 6 his tag.
Okay, I'm just wondering if where it is might actually be affecting whether or not that enzyme is going to function properly, because that may be telling you something about where the activity is. If the his tag stuff doesn't necessarily work the way as normal stuff does, is the question.
Mm-hmm. It's not near, I know that when we put the his tag on, it was in a specific position that shouldn't interact with other things of similar charge. So we did take that into account. I don't know the specifics of it, but I know we did definitely take that into account. So yeah, thank you. Great question.
We have one up here as well.
Well, it looks like your substrate might be auto-lysing. Is there a way to treat your substrate so that you can destroy the lysis, it auto-lysing? Is without anything, you're basically lowering your, without your ATP, you're lowering your peptidoglycan absorption. So I was wondering, have you treated the substrate ahead of time, like maybe taking the cells and boiled them or denatured them or something like that?
So this is our substrate specifically, so it's not changing in absorbance. Like these have a change, like this is 0. So that's the substrate specifically. Sometimes, I don't know if ATP is doing something or stuff like that, but yeah. Our substrate is staying flat like we would expect it to, but the other ones are doing a little bit of a weird thing. Yeah.
All right, if there's no more questions, let's move on to our next speakers. Thank you. You might just want to pass or Our next speakers we're going to have are from ATSU, the ATSU University. They're second-year DO students. I'm trying to steal Ohio State's thunder. So Cale Golden and Elise Curry will share this presentation out of the microbiology lab, I believe.
And physiology.
And physiology.
That's right.
All right.
Thank you.
Wait, how does this work? Oh, got it. All right. How do I go forward? Okay. Hi, everyone. My name is Elise Curry, and this is Cale Golden. And like Dr. Hauser said, we'll be presenting our research from Dr. Chamberlain in microbiology and Dr. Geisbuehler in physiology. So we specifically worked with Staphylococcus aureus, and a few characteristics about that is it is a gram-positive coccus, it is an inhabitant of normal flora, and it is resistant to a number of antibiotics. It does form biofilms, as you can see a nice picture of one here, specifically on medical devices. It does cause a number of cutaneous pathologies, endocarditis here, as you can see some vegetations growing on the mitral heart valve, and then some medical device infections as well. So many of you are familiar with bacterial resistance. We specifically work with bacterial persistence. So to differentiate between the two, bacterial resistance is the ability of bacteria to grow in the absence of antibiotic. This is inherited from one to another, and this does occur during active cell division. However, persistence is no bacterial growth in which the cells are able to lie dormant in the presence of antibiotic, and it is not inherited from one cell to the next. Again, there is no cell division with persistence. To further drive this point home, we have a few plates. Up at the top is the resistant cell. So as you can see here, we have some Staph aureus plated, and the red represents the resistant cell. After the antibiotic is added, only the resistant cell remains, and then when the antibiotic is removed, the resistant cells are then able to proliferate. At the bottom, we have the persistent cell. As you can see, there's some golden Staph aureus colonies with black representing the persistent cells. When the antibiotic is added, the bacterial-sensitive population is killed, and then when the antibiotic is removed, the persistent cells remain, however, are different because they're genetically different, and the bacterial-sensitive population is again then able to proliferate. So what is persistence-inducing factor? We discovered that this is an extracellular factor that Staphylococcus produces to induce persistence. Previous research, including Danny Hobtoo's, my master's work identified Piff from Staph aureus in SH1000, and my job was to partially characterize it. So I found that Piff was sensitive to boiling, sensitive to pHs greater than 8, and sensitive to protease K. The next step was to purify Piff, now that we completed the characterization. And the goal of this research is to understand what increases these persister numbers so eventually we can help eradicate them by awakening them and then successfully treating them with antibiotics. So the Piff assay that we use is what determines our activity levels. So we start with an overnight culture. We then take that overnight culture into 50 mils of TSB, and then we take 9 mils of that combination and add 1 syringe filter sterilized mil of Piff. We then grow to 0.5 optical density at 600 nanometers of light. We then perform a serial dilution and plate our colonies. We then count our countable colonies. And then with the remaining, we treat with 30 micrograms per mil of oxacillin, and we incubate for 16 to 18 hours. We then do a serial dilution and plate those colonies. So then we have our treated colonies and divide those by our untreated colonies and take that number times 100 to get a % survival. So one of the last things that I did with my master's research was to identify that Piff activity was less than 3,000 molecular weight. Now this took many attempts in which we finally found that adding arginine helped to disaggregate our protein, what we believe is protein. And we found that the greatest activity was then at filtrate and wash 1. So it was able to run through a 3,000 molecular weight cutoff filter at 2-hour centrifugation intervals. So then this allowed us to be able to set up the purification, which was the next step in our process.
Can I grab that from you? Yeah. All right, so like Elise said, the next step was purification. So we wanted to capitalize on the fact that Piff is less than 3,000 Daltons, and we spun the sample through a 3,000 Dalton filter and took out the larger proteins, and then we kept the smaller ones. After that, we wanted to further isolate the material, So we put it through a reverse phase column, which was a C18 column, and that is hydrophobic. The Piff bound onto the hydrophobic surface of the column, and then we eluted it and washed it with different concentrations of acetonitrile. We started at 10% and moved up to 70%, increasing by 10% increments each time. After that, we lyophilized each sample, and then we resuspended it and performed a Piff assay. So you guys are looking at the Piff assay. This is like the one that Elise explained earlier. And, sorry about that. So up in the left, you can see the negative control, and there's only a little bit of growth without the Piff activity. And then there's the positive control with the Piff right next to it, and there's quite a bit of growth. And then right here at 60% acetonitrile, you can see there's a lot of growth compared to the other washes. So we wanted to focus on that a little bit more. And also, we wanted to take this qualitative data and make it quantitative. So we looked at the percent survival. With the negative control, we're at 0.189%. With the positive, 3.35%. And then with the 60% acetonitrile, we're at 7.62%. So again, we wanted to focus in on that 60% acetonitrile. And we used high-pressure liquid chromatography this time to further isolate the possible Piff protein. You can see here there's 3 peaks of interest when we perform this type of column chromatography. One at about 13 minutes, one at 18 minutes, and another one at 39 minutes. So in future studies, we'd like to isolate these different proteins and perform a Piff assay and see if there's Piff activity in those. In conclusion, persistence-inducing factor activity was less than 3,000 molecular weight, like we said before. And then we performed the Piff isolation on a C18 column, which was eluted with 60% acetonitrile. And then lastly, after we put the 60% acetonitrile sample through HPLC column, we found that there were 3 points of interest. And I'd just like to thank Dr. Chamberlain and Dr. Geisbuehler for letting us work in their lab. So thank you.
We have time for a bunch of questions.
Oh, that's okay.
Put these guys on the spot. No.
Okay. You showed that and you mentioned that the Piff is an extracellular protein. That means it's not secreted by the bacteria. So where do the protein comes from? Is it from the broth or from the medium? Where do you get the protein, the Piff protein? And the other question is, when you showed the growth in the negative, it is 0.18%, but then once when the 60% acetylnitrile fraction is added, the growth is increased to 7 something. So it's several-fold increase. So whether it stimulates the growth also, apart from being a persistent inducing factor, whether it stimulates the growth of only this bacteria or any other bacteria, do you have any idea or comment?
Oh, I can answer yours. Go ahead.
So to answer the second part of your question, thank you. To answer the second part of your question, I'm actually not sure that Piff stimulates growth, but if you were to look at the negative control, there was already a little bit of growth, and that is because there may be some persisters that form their own colonies naturally, but the Piff acts as possibly like a cytokine, if you will, and it induces the cell to go into a sort of dormant metabolic state. And so it's no longer dividing, and therefore the oxacillin antibiotic that we treated it with won't be breaking down the cell wall as much. So that's the proposed mechanism that we're looking at this point. Does that answer your question?
Yeah, my second question.
Okay. Go ahead, Elisa.
Oh, super. Okay, so what was your question about why it's not an extracellular factor?
Extracellular factor, so where the factor comes from? If it is not from the secreted by the bacteria,
Yeah, I'm not sure.
where the factor comes from? Any idea?
No, so we're currently not sure where it comes from. We know that Staph aureus is making it, but we're not sure the exact mechanism that it's coming about.
So how do you know it's a protein?
So because of the protease K, it was sensitive to protease K treatment.
It could do anything. There is no conclusive way to say it's a protein. It could be anything, actually.
True, it still could. So further studies are needed.
So now that you show that it could be a cytokine or something, the only analogy I can draw is between the Piff is it's just like a hibernation in animals. They go into sleep during the winter time. Okay, so whether the same analogy holds good with the persistor because they go into sleep and they don't grow, and once when the right condition is provided, they start multiplying. Is it kind of similar to an hibernation in bacteria?
Yes, I would say yes. That's kind of the mechanism that we're going with, is that it does lie dormant, and that's what allows it to evade antibiotic treatment because the antibiotics target it during its cell replication phase typically.
Thank you.
One more question.
So going back to your graph at the very beginning where you were explaining resistance versus persistence, and you were talking about how the persistent bacteria, they just basically survive the antibiotic treatment, and then the bacteria can repopulate, so to speak. So has there been any kind of idea about why certain bacteria decide to take up this persistence-inducing factor versus the others?
Okay, so we said that these persistent cells were antibiotic tolerant. They weren't.
Right.
Okay, they weren't necessarily resistant to the antibiotic just
Right.
because they were in a non-dormant phase. They weren't able to be targeted by the antibiotic.
Correct.
And so was your question was what?
So then why, okay, so if you're saying that the persistence-inducing factor is an extracellular, or it's inducing persistence, right, in the bacteria, then I would think it would be present
Right.
there at the very beginning. So how come all of those bacteria are not persistent?
Okay. Yeah, go ahead.
Does that
Sure.
make sense?
Yeah,
Am
that's-
am I
Yes,
making sense?
That's
or-
a great question, and actually, that's something I asked to Dr. Chamberlain. Is this maybe temperature dependent, or does it depend on the availability of nutrients? And the way that I kind of like to look at it is that a few of the colonies out of the multiple that are formed kind of take the hit for the population, and they say, okay, if we're in a place now where there is enough room for growth, I'll let you guys grow, and then we'll stay dormant in case there is an antibiotic or some type of assault that comes through on the population, and so then they remain afterwards. I know that's kind of a surface level answer, but that's the way I understand it.
Who wants to have the lapel and who wants the hand mic? We've got a couple gentlemen here from Westminster College. They were here over the summer for the CRDP program, the Clinical Research Development Program. I will let them introduce themselves. I don't want to mess that one up.
Sounds good. All right, I'm Joshua Udinue. As he said, I'm from Westminster College. I'm a junior this year, and we had the pleasure of working in the AT Still Research Institute this summer for 2 months.
I'm Philip Udinue. As you can tell, we're twins, if you couldn't tell. We're undergrad students at Westminster College, like you said. We're juniors, both biochem majors. Looking at the pre-med track, we had a great time this summer thanks to Dr. Degenhart, which allowed us to do this research.
And then our research is about the contributions to postural alterations from a controlled ankle motion walker boot. So like some of the background, this image right here is what a general cam walking boot looks like. They're used for sports injuries. We've used them a lot on our campus for ankle sprains, and we've seen people wear them from around 1 week to almost up to a couple months, depending on the type of injury that they had. And these have started to be used a lot more than casts because of the ability of giving the patient to be a little bit more mobile than they would be without, or would be in a cast.
So with that, with that mobileness, we looked at how cam boot wear could create an unintended leg length discrepancy, resulting in secondary site pain in either the back, spine, or in the knee of the longer leg.
And one of our hypotheses was this picture on the lower left, and that is kind of an image of what the functional scoliosis looks like, and that's kind of what the body has to adapt to because of the leg length difference that the cam boot can cause. And so the purpose of the study was to analyze what happens immediately, and we looked at stationary posture and posture during gait. And our hypothesis was that the leg length inequality would be one of the major factors, and we looked at a couple different measurements. Those are the measurements listed. And then these are some images from the program that we used. So this is kind of what the program was looking for in its measurements. And pelvic obliquity and pelvic torsion were some of the ones we were focusing on the most. And so the equipment we used was the Deers Formetric 4D. It uses surface topography to create a 3D image of the spine and pelvis. So this is what it looks like in the image. It uses horizontal lines, and the computer actually measures the difference between the lines to create an image. And the greatest thing about this is that it's radiation-free. So we can do multiple scans in a short amount of time without any potential damage to the participants.
So the demographic range for the study was adults aged age 18 through 39 with no prior cam boot usage and with a BMI of less than 30. Because since the machine is using horizontal lights shown on the back, if your BMI is above a certain level, at some point the machine cannot pick up the distortion of those lights anymore, and it does not create an accurate picture.
And then we looked at 4 conditions. We wanted to kind of break down what the cam boot is doing to the body, and we will talk about those conditions in a second. So these kind of reflective strips right here are infrared markers, and those help the computer orient itself. So we placed those on the posterior superior iliac spine and on the C7 vertebrae, and we did that by palpation. And that helps it a lot with the dynamic studies to keep it oriented the whole time.
So the Deers system performs 2 types of scans. The first one is a static scan where the participant is just standing on the treadmill, the pedogate treadmill, and around, how many pictures are taken?
6.
6 pictures are taken in around a 12-second period, and then in the walking scan, around 300 pictures are taken in a 12-second period. So that allows us to create a picture of what's happening to the spine throughout the process of the gait.
And for this study, we used baseline scans and then used those to compare to the conditions that we did. So the first condition we looked at was the controlled ankle motion boot. This is what it looked like when the participant was standing on the pedogate treadmill, and we had them wear a shoe as well, because most people would be wearing a shoe while they wear the cam walking boot. We put all of our conditions onto the right leg. We just used that for consistency, and we used a random number generator to determine the order of when we were going to do the conditions. So that's what the cam boot looked like. This is what the heel lift looked like, and we determined how thick of a heel lift to use based on the difference in the thickness of the shoe that the participant brought in and the thickness of the heel of the cam boot size that they wore. And these are the ankle weights we used to simulate the weight of the boot. And then we used ankle tape to create the immobilization that the cam boot creates. So those are the 4 conditions we looked at to kind of isolate which condition was causing the most effect onto the body. And this is another summary of the population. And with this study and the Deer system, it creates a very large data table. It's thousands of columns long. So we are very thankful for our biostatistician who was able to do a lot of that work for us. And this is some of our results for the pelvic obliquity and pelvic tilt. This is, again, what the machine is measuring. And we saw an increase of 3.5 millimeters compared to baseline, which is what we were expecting to see, so we were happy to see that. And this is kind of what we were thinking of that would lead to the functional scoliosis. And as you can see, the cam boot on the static chart and the lift created a 1.6 millimeter difference, so not quite as much. And then the marker, tape, and weight did not create any change as we expected. This is for the dynamic data. The dynamic data has even more data points than the static, so that is going to take some extra time to analyze completely. But on this chart, every dot is a different participant, and this is comparing baseline to CAM. And as you can see, there's a positive correlation of an increase of the pelvic obliquity during the walking dynamic cycle.
So for pelvic torsion, the camera created a negative difference of 1.1 degrees compared to the baseline, and our p-value was where it needed to be significant for this study. And
we aren't entirely sure how this can affect the body. This is something that we'd want to look into further in the future and see what potentially could be the cause for secondary site pain because of pelvic torsion as well. But this is one of our other measurements that kind of came up as significant and we wanted to look at. This is the dynamic chart that we used for that. So as you can see, there is the negative correlation for this one. There is some outliers, so some of the participants had an increase, but that kind of happens for every study. There's going to be outliers for everything. And then in conclusion, the pelvic tip confirmed our hypothesis, and we were very excited to see that. One thing we were interested in seeing, this summer we were able to get 19 participants, and after that time, the apical deviation, which I showed in one of the first slides, so that's kind of the curve in the lower back, was actually showing up as significant. And then after Zane, who works in the research lab the whole time, he completed our study for us while we were starting the semester, and it kind of leveled it out and made it not significant anymore. So that kind of shows the importance of having many participants.
So it was much harder to get an accurate basis off of it with the dynamic data because we had around 80,000 columns. 8,000 column data points for each individual scan that we did. So beyond that, additional data analysis is necessary to fully understand what is happening with the immediate effects of the cam boot. And something that we're also looking at is the long term. We had a short-term hypothesis and a long-term hypothesis. We unfortunately did not get anybody for the long-term study because it's hard to find people who are just being put in a boot that you can study and track over the whole entire process that they are in their controlled ankle motion walking boot. And something that we are interested to look at that comes along with the DEER system is the pressure data. So we are going to be looking at how the ground reaction forces interact and could affect the secondary site pain with the peaks of ground forces changing with each of the interventions.
And this chart up here is kind of during the gait cycle how it changes from baseline to a cam boot. So there's some significant changes in pelvic tilt, which we expected, and we'd like to make some more charts and stuff based off that. And then one other thing we've noticed a lot of, you might have seen them too, is people will be using scooters with their controlled ankle motion boots. So we aren't sure how you'd be able to tell the difference between those, if those would create a different difference in leg length. But that was just something we wanted to mention because we've seen a lot more of those being used around our campus and in the world. And we'd like to give a big thank you to Dr. Degenhardt. He helped us a lot over the course of the study. And then these are the people that were in the research support lab that helped us the most over the summer. And then we'll open it up for questions now.
We got Dr. Snyder here, the Eric Snyder.
Thank you. Hey, for the pelvic tilt, any understanding on how that relates to known acceptable motions across the sacroiliac joint, and how much of that motion you were seeing was being accommodated by the sacroiliac joint, or whether it's kind of in predicted ranges of what you would expect for known motion across the sacroiliac joint?
We didn't look at the sacroiliac joint as specifically, but in the previous research that we read before we started this study, we saw that a difference of between 3 millimeters to 9 millimeters is when people started to notice some secondary site pain, and that difference in length is where there could potentially be an issue, and I believe it would be in that joint is where one of the biggest issues would be. But that's something else that we need to look at, is how much is too much and how much would create a difference that the body would have to adjust to over time and potentially create pain from that adjustment.
I missed whether you said your subjects were wearing the boot for injury, or if they were just normal subjects that put the boot on for the purpose of the study. And sort of if you have any comments to make on how injury might sort of affect this kind of study.
Yeah, so for our study, it was just the immediate effect, so everyone who was volunteered was their first time being put in the boot, so they had no prior injury. But the injury is something that we do want to look at in the future, which is why we have the long-term study there also. We're not sure how the injury and recovery process would affect the body as it's also adjusting to not only the injury that just happened, but also being put in a walking boot that is changing your leg length a significant amount.
Yeah, one thing that would be interesting is trying to find someone that we'd be able to get data on before an injury, and it's kind of hard to predict if a person's about to have an injury. So in a perfect world, we'd find someone, run through baselines without the injury, then the injury would happen, and then be placed in a cam boot and then track their progress and the changes in their body over the course of a couple of months.
You've got some extreme, your outliers, sometimes they were really very different than the others. I was wondering if you've ever checked to see if somebody who's an outlier, especially those extreme ones in the opposite direction, if they're an outlier in one, if they're an outlier in some of the others, and therefore they might have some difference in their function you're showing up just because they respond differently in multiple tests.
Yeah, that's something that we noticed as well. Like there would be some very extreme outliers, and that was something we'd be interested in studying more. During our time here, we were only here for 2 months. We were focused on getting as many people as we could because the goal was 40. But kind of slowing down a little bit and looking at seeing if someone has an outlier and seeing what could be leading to that outlier and possibly either disqualifying from the study because of that outlier or something like that would definitely be interesting to do in the future. Does that answer your question? Oh, yeah. Most of the time, if someone was an outlier in one of the categories, they'd be an outlier in the other categories as well. Yeah, potentially. But we... yeah, exactly. Yeah.
Guys, first of all, kudos. You guys spent only a few weeks, but you guys are answering amazing. So I don't know your background, but you guys are tackling the question very well. That's a good thing. Okay, my question is, you conducted the study with the normal person, right? And though we know that people who have scoliosis, you put the cam boot either in 1 foot or, I don't know, that's the way it happened, to adjust their gait. But then you are conducting the study with the normal person, and there, what I feel for your baseline study, you just put the boot in only 1 foot. I saw on the right foot, right? And then you study. But as such, even in the normal person, if you see the one with the boot and with the normal foot, there is a difference. That's going to cause some changes in the posture, okay? So why don't you guys do where they wear the boot in both the leg and then see just by wearing the boot, whether it changes the posture. And then you can change the leg, either right leg or left leg. So now the one with the boot on the both leg or both foot, that should serve as a baseline. So just by wearing the boot whether it has any effect on the posture. And then you can change change it either with the right leg or the left leg.
Yeah.
So what's your comment on that?
With that being said, the having both people or having both legs in the boot would almost be the same as having neither of them on, because you're still looking at just where the hips are normally before that. But that is something that we could look at for future studies.
Yeah, that'd be interesting to look at, because most of the boots have like a rocker bottom design, so it's kind of forcing you to walk a lot more heel to toe than you usually do. So having two boots on, which I've never seen before and we never really thought about, so that's an interesting thing to look at. Would be interesting to see how that would affect like the dynamic data as well. So that'd be something interesting to look at in the future.
Thank you.
Thank you, guys. All right, our next speaker, Nathan Needham, one of our very own KCOM. What year are you? 2019 graduate. So he's in the Department of Anesthesiology, coming to us from Northeast Regional Medical Center.
Is there a laser pointer? Yep. Okay. Perfect. All right, well, again, my name is Nate Needham, first-year resident with the Anesthesia Department at Northeast Regional Medical Center. And I had a really interesting patient that I took care of this last year that I'd like to share with you. So this is the way that the patient that was presented to me. 52-year-old, he was a 58-year-old with a 52-pack year smoking history, hemoptysis for the past 6 months, unintentional 40-pound weight loss over the last 3 months, and a history of a lung nodule on previous chest CT that had never been followed up before. So obviously this patient is non-compliant. He did not like to go to the hospital. He did not like doctors. So if any of you ever see this question on boards, what do you think the answer is going to be? So this confused the picture a lot for multidiscipline, for several different physicians that were overlooking this patient. So as we can see here, and one thing, this picture also delayed the definitive diagnosis for this patient. So what we can see here is this patient is hemodynamically unstable. And the most important thing in patient care is treating what's going to kill them right now. And suspected lung cancer, that's kind of, yeah, this patient's really sick. Something's really wrong with this patient. But he came in after receiving a bolus of fluid. We can't get his pressure up. He's tachycardic, elevated heart rate, fever, white count that's extremely high. And he's going into respiratory failure, kidney failure, liver failure, multi-organ failure. SIRS positive, which is an indicator of sepsis. He was positive for every single one of them. Shock, multi-organ failure. This patient has septic shock. But where is this infection? We, on his previous workup, the radiologist knew the history of this patient. And the report that we got from the radiologist said that the patient had normal lung anatomy for a person that has been smoking with a 52-pack year smoking history and per the patient's age. Everything else was generally normal. So what we did with this patient is we took this patient directly into the ICU, and that's where I took control, or I was involved with this patient's care. We needed to start this patient on vasopressors, get the pressure up, and an arterial line was placed. From the waveforms provided on the arterial line, large blood pressure variations were seen as described as Pulsus Paradoxus. So as you can see here, this is the arterial line here, and you can see this kind of peaks and valleys, this wave that was going on with his arterial line pressures. This can also be seen on pulse oximetry because pressure is associated with perfusion of the finger if your pulse ox is on the finger. And you can also see that these valleys are associated with these valleys here on this end-tidal CO2 monitor, and those are inspirations. So Pulsus Paradoxus is described as blood pressure variations greater than 10 millimeters of mercury between inspiration and expiration. So again, just pointing this out again, this valley is associated with this inspiration. So going to kind of go over the physiology of what's been going on, but once we figured this out, we had this patient that was given to us. We thought lung cancer. Everybody thought lung cancer. This patient's pretty much done for. We could have just let the patient go to oncology. But like I said, the chest CT was read normal, but we did not know where this source of infection was coming from. But with this information that we received from this arterial line, we knew that there was a physiologic change that was happening to the heart, and we didn't know what was going on. So we pulled up that CT image that was read normal, and because there's a little bit of clouding on the history, we eventually saw this large pericardial effusion. Along with that pericardial effusion, if you go a little bit higher above the heart, there was a large abscess that was above the heart that was communicating with the pericardium. And the radiologist could, with the density of the fluid within the pericardium, he suspected that that fluid was pus. So, everybody, I want you to take a deep breath because we're going to talk a little bit about physiology. Now really, everybody taking a big deep breath. What's happening when you take a big deep breath in? Your diaphragm's dropping, flattening out, your chest wall is expanding, causing an increase in intrathoracic pressure, which draws air into your lungs. Along with this normal physiology, an increase of venous blood flow comes into the right side of the heart. This is called your thoracic pump. This is normal physiology. When this happens, an increase in volume in the right side of the heart causes the ventricular septum to bow into the left side of the heart, into the left ventricle. This causes the left ventricle to fill less, causing less stroke volume and a drop in blood pressure, a drop in, like I said, stroke volume. So I know this looks confusing, but now on top of that, we have an abnormality. We have a pericardial effusion which is compressing down on every side of this heart, plus we have this normal physiology happening when we take a deep breath in, bowing this over more, and definitively giving us the definition of Pulsus Paradoxus, where it's greater than 10 millimeters of mercury between inspiration and expiration. So we could see this level here, around like 110, and this one up here, it's kind of hard to see, but 125, 130. So that for sure gives us this diagnosis. So we contacted and consulted cardiothoracic surgery. An immediate pericardiocentesis was performed. And usually this is done under ultrasound guidance, but I thought this image was kind of interesting. You can see this needle coming in on CT guidance right into the pericardial sac to be able to extract that. And 800 cc's, almost a liter of pus, was drawn off of the pericardium of this patient. Immediate hemodynamic stability was achieved, so the patient was stable now. But something that was really interesting, I gave you the answer. So the next morning I went and saw this patient. He was doing a lot better. He had received his color back. I had never seen a patient so white in my life because of how low his pressure was. His face was literally as white as this paper. But he had this history of hemoptysis. And he had a whole cup with him that was full of blood. And per nursing, he had been coughing up blood all night. And so I went in there to see the patient, and I wanted to see what was actually happening. And he was just really spitting up this blood. And so I think there was a mixed picture between hemoptysis and really just like hematemesis, like spitting up blood. And so we ordered an EGD, and this EGD, this is abnormal, but this EGD showed severe necrosis of the esophagus with suspected, what's the term? Penetration, a tear in his esophagus that we suspected that was communicating with that mediastinal abscess. So in conclusion, the patient suffered from an esophageal perforation, which led to a mediastinal abscess. This abscess eroded into the pericardial sac, causing a pleuritic pericardial effusion. The treatment for a pleuritic pericardial effusion is immediate empiric antibiotic therapy. Pericardiocentesis should be performed, followed by a subxiphoid pericardiotomy if needed. So I hate memorizing long lists of things. So that's why I went through the physiology to understand what's going on. So again, if the heart's compressed, not working as well, if the muscle of the heart's not functioning as well, if you block blood flow back to the heart, any of the following are going to be associated with Pulsus Paradoxus. So we can go through this list, like what we had talked about, constricted pericarditis, pulmonary embolism, and you can read that list there. But yeah, that is my presentation. Do I have any questions?
This is a very interesting case. Yes. So for this case, what we can do better for this patient? So patient admitted for like septic shock, right? Blood pressure low, and we see it from the temporal knot also. So I don't know, like with the temporal knot like this, and they have the variation on the arterial line pressure, I think flux is the most important at that time, because the volume deplete preload. So we need to give like flux like not only 30 ml per kilo, we need to give more for that before we can start the vasopressor. That's the first choice, first thing. Second thing, for this patient, do you have any like tail leak EKG for this patient? Because with the temporal knot, sometimes we can see the site of temporal knot, like electrical alternance on the EKG tail list. Yeah. We can see for that. And
so you don't see that on here, but what he's talking about with electric alternans is you can see a larger peak on one, and then the next one shorter. And so that's another indicator that you can see on EKG if you happen to get that.
Like the hot swing inside of the pericardium. And with the, when we do the pericardiocentesis, if we see that the pulse, that means this tapping is not enough. The patient may need like window or training, because the pulse, that gonna, the septic shock not gonna be improved with training. That's I think the second step for the patient. Yeah, thank you.
Okay, just mostly for my own curiosity, do we know what was cultured out of that?
So so once we got to this point, we knew that this patient was going to have, like I said, this subxiphoid pericardiotomy. They'd have to have heart surgery.
Right.
Then when we found out about the esophagus, they'd have to have an esophagectomy and an esophageal pull-up. So at this time, the patient was transferred, and I discontinued.
I was just curious if you knew, because I was like, wow.
Yeah.
That was impressive.
Yeah.
So.
How's this guy doing? Is he still alive?
Honestly, I hope not. He was very sick, and from that surgery, the mortality is extremely high. So.
On the CT, was there a comparison just with the previous lung nodule had progressed?
So that's what was interesting, is the lung nodule wasn't there on chest CT. That's why we were confused at the beginning. The radiologist report, lung nodule no longer there, lungs look normal, everything looks pretty normal. So his intervention took longer because of this confused picture of lung cancer.
Was he eating over this past 6 months?
He was. Yeah, it was surprising how sick he was and how he could have waited that long to come to the hospital or the ER because he was extremely sick.
On the timing, when the lung nodule, I think the timing and where it was, just wondering if maybe that was sort of a bulge out of the esophagus that was getting towards the pericardium rather than a lung nodule.
Yeah.
I mean, it depends on where that, I mean, it might have been nowhere near. It depends where that was. That might be an interesting thing to find out.
Yeah. Okay.
All right. Thank you. All right, our next speaker is Lily Mers. She's from the Truman State University, Department of Exercise Science. Under what, the direction of Dr. Mayhew? Is he here today? No? He's close to retirement, isn't he? And Michael Bird as well. He's already retired several times. Yeah.
Let's try again. Step rate is the frequency of steps, so a left foot to a right foot, given over a given unit of time. And previous research has shown that as running speed increases, step rate also tends to increase, coincidentally. And we also looked at step rate, which is commonly divided by height and known as relative step length, as that's SL divided by HT here. And previous research has found that as running speed increases, step rate or step length also tends to increase to reach about a 1-to-1 ratio, saying that at fastest speeds, step length is often equivalent to the runner's height. That division by height helps normalize the values to control for differences of participants. And we were curious as to if Footstrike pattern has an effect on running speed and these other characteristics. So we have done previous research, and we use these 3 Footstrike patterns as options. Okay. Okay. There we go. So a heel Footstrike pattern is when walking or running. In this case, the heel is the first part of your foot to hit the ground. And then we go to a midfoot Footstrike pattern, which is when your foot lands completely at the same time. All parts of your foot land at the same time. Or a forefoot Footstrike pattern, which is when you run and the first part that hits the ground is like the balls of your feet. So not necessarily running on your toes, but it's just that's the first part of your foot that lands. There has been previous research on professional runners, and they have found that between those studies, groups of professional athletes typically tend to have the same Footstrike patterns. For example, ultramarathon runners all have that heel Footstrike pattern. Oh, wow, I'm a mess with this, sorry. They tend to have that heel Footstrike pattern, but as many of us are not elite ultramarathon runners, we have looked at a more heterogeneous population to see how these interactions occur. Which leads us to our purpose. We assess the gait differences in runners and non-runners of a heterogeneous population to determine if training has an influence on step rate, step length, or Footstrike pattern across slow to sprint speeds. In this study, we had 190 participants all self-classify themselves as either runners or non-runners, and they ran along a track similar to this one here, 4 times each at self-selected paces, categorized as slow, medium, fast, and sprint. They were given the acceleration zone to reach their ideal velocity, and then were told to maintain that velocity consistently through the measurement zone. While they were in the measurement zone, their time was recorded electronically, and we also recorded step rate, step length, and foot strike pattern. Foot strike pattern was determined through a small group consensus of trained individuals. We video recorded 57 of our participants and analyzed their foot strike pattern on the computer to determine our validity of proposed foot strike pattern, and we were correct for over 90% of our subjects, which is higher than most other research projects of this caliber. And these graphs over here, we have step rate versus running speed and step length versus running speed. That is all of our data plotted, and you can see the general trends are as expected due to previous research. Here we have running speed across the 4 self-selected paces. And these red circles here are showing the difference between men and women. And we see that men are significantly faster than women at all 4 paces. And these green circles represent runners and non-runners. And we can see that runners were significantly faster than non-runners at the fast and sprint paces. And also at sprint pace, we have a strong interaction effect, saying that the differences between men, women, runners, and non-runners all played a role with the data diverging here. Next, we have step rate across the 4 self-selected paces. And once again, that red circle is showing that men have a higher step rate turnover than women, but only at the sprint speeds. And also at sprint speeds, we can see that runners have a higher step rate turnover than non-runners. And there are interaction effects at the medium, fast, and sprint paces, so we can see that differences between runners, non-runners, men, and women begin as early as the medium paces. Next, we have relative step length across the 4 self-selected paces. And we can see that men have a higher relative step length compared to women beginning at fast speeds. So men are more likely to reach this 1-to-1 ratio before women are, which was that ideal ratio. And now we have runners have a longer relative step length than non-runners. They are quicker to achieve that 1-to-1 ratio and often exceed the 1-to-1 ratio compared to non-runners. And there are no interaction effects at the relative step length. So here is our total data for foot strike patterns across the speeds. And as predicted, running as speeds increase, speeds tend to move from a heel strike to a forefoot strike pattern. As was confirmed with our study, we see that it's that peach color at the top, the growth of forefoot foot strike patterns as speed increases. So this was as expected. And we are comparing men and women's foot strike patterns across the running speeds. So once again, we can see the growth in forefoot foot strike patterns across all the speeds. However, we see that 60% of men were forefoot foot strikers at the sprint speed, while only 31% of women were forefoot foot strike patterns at the sprint speed. Here we have runners and non-runners across the speeds. Once again, it follows that pattern of increasing forefoot foot strike prevalence. But however, 58% of runners use a forefoot foot strike pattern, and only 35% of non-runners use the forefoot foot strike pattern. And in conclusion, when comparing men and women, the men were faster than the women at all speeds. However, this increase in speed was not due to solely step length or step rate. It was a combination of the two. And men were more likely to achieve that 1-to-1 ratio at sprint speeds, or at fast speeds, which we found that men may be more likely to increase their step length before they increase their step rate as they are running sprints. And men had more prevalence of forefoot foot strike patterns at sprint paces, but in the slower paces, the differences in foot strike pattern were very negligible. And comparing runners to non-runners, runners had a significantly larger step length pattern at the fast and sprint paces. And also at sprint paces, practice runners had a higher step rate. So practice runners had higher step rates and step lengths at the sprint speeds. And runners had more forefoot footstrike patterns than non-runners at the fast and sprint paces, and the slower paces were also negligible differences. So overall, there are true biomechanical differences between runners, non-runners, women, and men, but there are also physiological differences there that need to be accounted for. And future research will need to evaluate whether these effects are more innate or can they be trained or perhaps formally coached. And the men and women, as well as runners and non-runners, do differ with respect to basic gait characteristics. And thank you all for listening to my presentation. I will be at poster 413 later, but I think we have some time now for some questions.
I was wondering, you said you found a difference between the men and women, but did you use that as a covariate when you analyzed runners versus non-runners?
We did both, so I'm going to go back here, maybe. So yeah, there are differences between the men and women and the runners and non-runners. I'm not too sure about the statistics and how they were ran, but we did cross a 2x2 multivariate ANOVA to look at the differences there.
So I'm really interesting work. I'm curious why, just your thoughts on it, why we saw more women sprinters doing the midfoot strike as opposed to the forefoot strike that we always saw with the men sprinters.
Yes, thank you for your compliment. We are not really sure. I could only speculate maybe differences in like shoe preference on a day-to-day basis or perhaps the anatomy of like the pelvic structure, but further research will have to be done to analyze that.
Thanks for the presentation. On the foot strike characteristics, I'm curious how you measured that, and then so between the forefoot, the midfoot, and the heel strike, and how reliable you thought that data was, or how you went about doing that.
Yes, so we had a group consensus of trained individuals for this study. So it wasn't just one person watching it, but I definitely agree there is a lot of gray area what distinguishes a midfoot foot strike pattern from a forefoot foot strike pattern. At that point, we just, we would discuss within the group, and then we also had the video recording, so figured we were 90% accurate for our observations. But yeah, there is a lot of gray area in between those.
Were you blinded to the groups they were in as you were having the discussion?
We were not. There's what?
They didn't know if they were runners or not.
Oh yes, we didn't know if they were runners. We were, yes, sorry.
They didn't tell if they were male or female.
Yes.
But the video could have even been done, right, the video you couldn't tell if they're runners or non-runners, and it was focused on the feet, but they had to identify them with the previous day.
Yes, it was partial blinding. Yes. It was a mic. Yes.
How were the 4 speeds defined?
They were slow, medium, fast, and sprint, so they ran at progressive speeds. So they all started with slow, medium, fast, and then grew to sprint. But the 3 slower of the speeds, it was all kind of open to interpretation, so we don't really know, was your medium equivalent to my medium? But we do know for sure that everyone's sprints were truly their 100 max sprints.
Okay, your study shows the general difference between male and female, and that's what males scored high in all the categories wherever you analyze. So now my question is, what makes this difference? Did you try to correlate it with their physique or muscle strength or their mental ability or the psych? So do you find any correlation that why they are superior than, not superior, they perform better than female in all the categories? Are you trying to do, or have you tried to correlate it with their physique, their built structure, musculature, or their gait, or their psych, their mental ability?
I think it is definitely a combination of the anatomy, physiology, and the psychology there of causing men to have faster speeds. But in some of these studies, such as this one right here, so we can see that women have a longer step rank. Women runners have longer step lengths than men non-runners. So there's not a complete difference in physiology here between men and women. It also has to do with athletic background.
While the next one comes up, let me ask you, how did you define who's a runner?
Yes, so they were self-proclaimed runners or non-runners. We advise them to self-proclaim themselves as a runner if running was a regular part of their workout routine.
Thank you.
So there will be more opportunities for questions for all of the presenters during the poster session. So make sure that you do take some time to talk with the people who present this morning in the afternoon session as well for any lingering questions.
All right, our next speaker is Lewis Wood. He is a second-year medical student working with Dr. Breshew and Dr. Kondrashova. All right, take it away.
Is it working yet? Sweet. Thank you, Dr. Hauser. Good morning, everyone. Today we are going to be talking about blood pressure responses and cardiac structural changes. So blood pressure is maintained or elevated with muscular work. This is called pressor reflex. It maintains that through vascular conductance or generalized sympathetic activation. It has been found that exercise training attenuates pressor reflex in already healthy individuals. And it also has been noted that it can counteract exaggerated pressor reflex in cardiovascular disease. Repeated and prolonged, it's a well-known fact, repeated or prolonged elevation in blood pressure can lead to concentric hypertrophy. What is relatively interesting is that in athletes, this phenotype, concentric hypertrophy, is considered sometimes an advantage, a good thing, but in the setting of chronic hypertension or heart failure, the same phenotype is considered a disadvantage and a bad quality. So we wanted to investigate the difference between blood pressure response to isometric contractions and left ventricular mass. Our experimental model so far has involved 25 healthy men and women. They came in on 2 separate days for testing. The first day, we measured their left ventricular mass. We did that with an echocardiogram in M mode. The echo was measured at rest, and we did it with, or we calculated the mass with these dimensions here. The second day, they came in for a single sustained isometric bout. They did so at 30% of their maximum voluntary contraction. They maintained their bout until volitional exhaustion or until they were unable to maintain that 30% target force. We measured their force with a dynamometer, and perceived exertion was taken every minute of the bout with the Borg CR10 scale. Surface EMG was taken for the flexor carpi radialis, flexor carpi ulnaris, and flexor digitorum profundus. Blood pressure was taken at 1-minute intervals along with the perceived exertion, and it was taken with an automatic cuff. And lead 2 ECG was collected continuously throughout the bout. We found that maximum contraction was greater in men than in women. We found that when the maximum contraction was corrected for forearm strength, it was still greater in men than in women. And 30% of 28.5 and 30% of 19.2 is still greater for bout force in men than in women. Left ventricular mass and left ventricular mass index were both found to be greater in men than in women. Resting systolic blood pressure was greater in men than women. However, diastolic resting blood pressure and resting heart rate were found to be similar among the two. So resting systolic, resting heart rate, and diastolic. Blood pressure response to the contraction bout was seen to be similar between men and women, as you can see in these two rows here. So this is our entire data set. Every one of these horizontal lines represents one of our participants' bouts. This measures their force at 30% of the bout force over time to exhaustion. You can see some of these variations here. That's if they were unable to maintain that target force, we went ahead and cut them off. Something interesting to note from this graph is we found that the maximum contraction is inversely related to the time to exhaustion as the graph kind of goes down. The next few slides, I'll talk about our normalized EMG data. We calculated normalized EMG by taking the EMG at each time and dividing it by the maximum EMG reading of the day. So for this graph, this shows that flexor carpi ulnaris has increased muscle activation over time of the bout. You can kind of see some of these individual participants here, that the muscle activation increases until exhaustion and then kind of goes out, and then this 10-minute that just keeps going out. Flexor carpi radialis shows a very similar pattern. And you can see some of these individuals kind of increase here. Muscle activation increases over time. Flexor digitorum profundus shows a similar pattern with slightly different variation. So all relatively similar muscle activation increases over time. This graph shows that there's a relatively strong correlation between systolic blood pressure and muscle activation. So as muscle activation is increasing, so is your systolic blood pressure throughout the bout. And this graph shows very similar with the perceived exertion. So as muscle activation increases with the EMG, so does your perceived exertion, with a relatively strong correlation as well. Left ventricular mass was found to have a moderate correlation with the change in systolic blood pressure. There was a little bit stronger correlation at 2 minutes than there was at 1 minute. So in conclusion, we found that blood pressure and heart rate response to isometric contractions was similar between men and women. We found that initial blood pressure response appears to be related to relative tension, but changes over time are related to increased central command and motor drive. Blood pressure response does not appear to be related to differences in maximum contraction or baseline blood pressure, and there is a moderate relationship between systolic blood pressure during contractions and left ventricular mass. So thank you for your attention, your time, and I'd like to open the floor to questions.
Yeah, this is interesting presentation. So do you use any statistics and you calculate the p-value for any of in your presentation? Because p-value is very important too.
For correlations, yeah, we did not yet do statistical analysis. Since we only have the 25 individuals in so far, we'll probably do a little bit more statistics as time goes on.
So you found that muscle activity, electrical activity went up, but the force generated was the same. So how do you explain an increase in muscle activity but a sustained force?
That could be so muscle fibers will start to more will be recruited over time, which can start to create more muscle activation, which maybe is what the EMG is picking up, even though the force is staying the same. And as the participants are going to contraction or to fatigue, to exhaustion, more activation will start to occur, increasing your EMG potentially. Does that answer your question?
Okay. My question is, what kind of task you ask them to perform? Because it's isometric contraction, meaning that the muscle maintain the same length, right? It's not an isotonic contraction, so isometric contraction. So what kind of task you ask the participants to perform to make the measurements?
So they had their arm at 90, and they had the hand grip, and they had their hand. They squeezed their hand to 30% of their maximum
Squeeze
contraction. Squeeze the dynamometer.
Okay.
Guys letting them off easy. No, just kidding. Let me ask you a question. Why the 3 muscles? Why those 3?
So the 3 muscles are your primary flexors of your hand. So we wanted to measure those in to get the measurement.
Is there any reason to do 3 and not just 1? Is there a primary difference in some characteristic of them that you would want to investigate?
Maybe later we would like to look at the different characteristics of muscles, because with the profundus had a little bit different pattern, we think maybe it might be a slow twitch. Later we can kind of figure out different characteristics between the 3 of them later on. But for now we haven't started looking into that yet.
All right. Oh, looks like we got another.
So in the beginning of the lecture, you kind of talked about how left ventricular hypertrophy can be advantageous in athletes, whereas it can be pathological in other cases. Were you trying to correlate that at all with this research, or were you just kind of giving background on that? Because I thought that was super interesting.
A little bit of background. We also think it's very interesting. So we've only tested healthy individuals so far, and I think eventually we want to get into testing heart failure patients and then comparing the 2 later and seeing what the difference would be there.
All right, thank you.
All right, thank you very much. Okay, our next speaker is coming to us from Truman State University, Dr. Sempertaghi. She's in the Department of Biology.
Pocket, is that okay? Hello. Thanks. Yeah, it works. Good morning. Thank you for having me here. Today I'm going to be just a presenter because most of the work done in this project was performed by one of my students over the summer. So this is a summer project we did in our lab. Just as a little bit of a background, we know that Pseudomonas aeruginosa is the most common gram-negative bacterium that is causative of nosocomial infections. And this has been implicated in many cases of urinary tract infections as well as septicemia. Now, it has been found also to form resistant biofilms that are very difficult to remove in the hospital setting, and also they release toxins like pyocyanin that are thought to play an important role in the virulence of this bacterium, although the role is not defined yet. Now, previous studies have found Pseudomonas aeruginosa in used sponges in hospitals, mouthwash swabs, surgical equipment, and also in hands of the healthcare workers. So in the US, we know that 1 out of 25 patients that go to a hospital suffer a nosocomial infection, and out of these patients, 27% have been infected with Pseudomonas aeruginosa, and raising the cost of healthcare to about $20 billion as nosocomial infection in total. Also, previous studies have found that when trying to remove Pseudomonas aeruginosa from a surface, the surface material actually altered the bactericidal effectiveness or activity of several chemicals that are commonly used. And common disinfectants also are less effective when the bacteria are forming biofilms or they are attaching to surfaces. Therefore, they have raised the importance of looking for alternatives to remove or to eliminate Pseudomonas aeruginosa or these bacteria from the environment. Delibiribrio bacteriovirus is one of the new possibilities that has emerged as a biological control agent. This is a predatory gram-negative bacterium. There are many predatory bacteria found in the environment, but this is one of the, probably the most studied so far. That is a multiflagellated microorganism found in the sewage, found in the soil, and even found in the human gut. These bacteria, they preys on other gram-negative bacteria and some gram-positive bacteria too. And many studies have already been looking into the possibility of using this particular bacterium as a potential antibiotic for treating infections in humans. So you can see here a little bit of the life cycle of this very peculiar microorganism that usually attaches and invades the intracellular, the pathogen, the host bacteria, and forms what is called a veloplast that it starts dividing in a different style, which is called a non-binary fission, which is unusual for bacteria. This is one of the unusual means of division. And then the bacteria, once they have divided, they are released into the environment. So our objectives were to compare the effectiveness of Delibiribrio bacteriovirus and compare that effectiveness to the effectiveness of two chemical control agents very often used in the hospital setting, which are Clorox and Lysol, and also testing the capacity of Delibiribrio bacteriovirus in reducing the number of Pseudomonas aeruginosa in various surfaces that can be found in the hospital setting. So what we did is we prepared different solutions of the chemical agents that we wanted to compare, and we diluted them to the advertised or by the recommended concentration by the manufacturer. And we prepare a solution of Delibiribrio bacteriovirus to 100 plate-forming units per milliliter. And we use water and lysate of the bacteria as negative controls. We added them to a known concentration of Pseudomonas aeruginosa, and we incubated, or we let them sit for about 30 minutes at room temperature. After these 30 minutes, we plated these the cultures, and we incubated the plates to let the surviving bacteria grow, and we conducted colony counts after that. We conducted 10 replicates of each one of the experiments to gain statistical significance. In addition, we tested 4 different surfaces, and we added the same Pseudomonas aeruginosa suspension. We added 0.5 milliliter of it to the surfaces, and we inoculated them. We let them dry. And after drying out, we applied the 4 different treatments, and we did replicates of also for these experiments. After the treatment for 30 minutes, then we sampled again, we swabbed again the surfaces, and we plated those microorganisms and incubated them for 24 hours and conducted counting of the surviving bacteria. So what we found is that in suspension, the bacteria were mostly reduced by the Vibrio Bacteriovirus. So this is just as a visualization of the number of colonies that survived the treatments. So we had less colonies surviving with Vibrio Bacteriovirus, and the least effective of the chemical treatments will be Lysol in suspension. When we looked at the results for the surfaces, we found out that the Vibrio Bacteriovirus compares or is very similar to Lysol in terms of reducing the number of bacteria in different surfaces. And Clorox was not as effective in our experiment, and this variability also was shown among the different surfaces we had. And when we tested, when we compared and our significance, we found out that actually there was a significant reduction of the number of bacteria using the Vibrio versus Clorox in the head wall type of material, as well as the tile. And also we had a significant reduction of Pseudomonas aeruginosa using the Vibrio versus Lysol. We didn't find any significance in the other materials and the other combinations, which was interesting for us to see. But we could conclude that the Vibrio Bacteriovirus is more effective than Clorox and Lysol in reducing the number of P. aeruginosa from rough surfaces, and only more effective than Clorox in smooth surfaces as tile. Now, we find a potential of this bacterium as an antimicrobial agent, but the type of surface needs to be taken into account, and as well as the environmental conditions. Now, something that is a limitation for us is that most of the Vibrio Bacteriovirus are obligate predators, so they need to feed on a host bacterium. However, there is a mechanism that they can follow, or there is a mutation that may happen that allows the bacteria grow extracellularly. They can multiply outside the host when they mutate, but that is a rare case. So that will be a limitation of our project, because we counted the colonies that grew, and we are not absolutely sure that those rare cases didn't happen in our study. And finally, obviously, we will need more replicates, more than 10, to gain better statistical significance. We would like to test more surfaces and other types of environmental conditions, like lower pH or higher pH solutions and other materials, as I mentioned. So I would like to thank overwhelmingly my student, Bria Berry, who was the person who conducted this research, and without her, this wouldn't have been possible. If she hadn't graduated, she will be presenting here. And also the Department of Biology at Truman State University and the Office of Student Research, who funded the project during the summer. So, friends, thank you very much.
Ooh, we got a lot of them. I'm going to just start here in front.
Yeah.
I just have two quick questions. One, have you considered a comparison to something like chlorhexidine or betadine that are used, in a hospital setting, surgical setting for?
Yes, we have. We have considered. We chose sort of like the most widely used for a start, but yes, we have considered using those.
The quick second question is: do you think that biologic agents like this would scale well from a cost perspective compared to chemical agents?
That is an excellent question. They are found environmentally, but obviously the cost will be to produce them and modify them to be effectively used. I'm not sure, honestly, but yeah, that is a good question.
Yeah, I have a couple questions. So, B bacterial resistance classified like virus or bacterial?
It's a bacteria.
Bacterial. Yeah. So, any potential like you can call like, because if we use the bio and antibiotic, any potential can be harm to human being because it's a bacteria, right? So I don't know.
Well, actually, because the Vibrio bacteriovirus has been found in our gut, and normally it is not considered a pathogenic bacterium yet. And also, there are some scientists who are modifying the bacteria to try to make it work, make it target just specific pathogenic bacterium and not affecting the host. But there are already studies on that.
Yeah. Thank you. Yeah.
So the concentration right here. So the concentrations that you used, you said they are stated were stated from the manufacturer, right? To probably make it safe in the household use. But how does it compare the concentrations to hospital settings? I mean, probably there you have more of the bacteria, possibly, and you use different concentrations. And I guess concentration is also a big factor of how you can kill the bacteria.
Yeah, that is a very good point. Yeah, we use the recommended concentration by the manufacturer, but yeah, we honestly didn't look into the concentration used by hospital settings. But we actually assumed that it was the same, but it's possibly that it's not. Yeah.
So Carolina, was your bacterium multi-drug resistant like you might find in hospital?
No, our strain was a non-pathogenic. It was an ATC strain that is not multi-drug resistant. Yeah. Mm-hmm.
Okay. My question related to this, the question from this gentleman as well as from Dr. Easterwood. So basically, like you are using another bacteria, Bacteriobus, okay, B. Bacteriobus. So what is the basic nature of that, whether it has its own effect, just like any other organism? And number two, have you tried with other bacterial strains such as Staphylococcus aureus? That's also spread on the surface. So what is its effects on the other bacterial strain?
Okay, so for the second question that you asked, it can affect also Staphylococcus aureus, and that is actually one of the gram-positive bacteria that it can affect. But the difference is that in gram-negative bacteria, they tend to feed by being internalized in the host. In gram-positive bacteria, the feeding or the predation is different. They feed from the outside of the cell. So I'm not sure exactly how effective will be then in gram-positive bacteria because of the difference in the way of they prey on those bacteria, but it is certainly one of the preys for them. Would you mind repeating the first question, please?
The basic nature of the bacteria and whether it is an virulent type or non-infectious type, or what is the nature of that as such. And then I just want to echo what Dr. Easterwood said, whether we can accept this as a biological control to use in the clinic. That's another thing. So what is the basic nature of this one, so which you think that it can be used as a biological control to be used in the clinic? So first of all, whether we can use this bacteria as a biological control, whether we will allow, as Dr. Easterwood raised the point. Mm-hmm. So my question, what is the basic nature as a bacteria? This is also a bacterial organism. Mm-hmm. So it has its own effect and all. So what is its basic nature, that whether it's infectious or virulent or?
Okay, yeah, so it is not known to be a human pathogen. It is an environmental bacterium that predates only on other bacteria. So preys on other bacteria, sorry. That is the main nature of that. Now, as the potential of an antimicrobial, scientists have seen that it has potential because you can treat multi-resistant bacterium with this antibiotic multi-resistant bacterium with these predators, and then easily clear out the new bacterium because these are not multi-resistant. Yeah.
Did you ever look at the Pseudomonas that survive and see, are they resistant to the predatory bacteria's attacks, or were they just lucky?
No, I didn't. I didn't look at that further. Yeah, we didn't look at that further. Thank you, but it's a good thing to continue for continuation. Yeah.
How did the concentrations that you used of your bacteria compare to the concentrations that you find in the gut? And if this were to be something widely used, how do you think that would eventually affect the gut microbiota system?
That is a very good question. Yeah, I think if they were using this bacterium as a means of treatment, they will have to modify it somehow to target only specific bacteria in order not to attack others. Because actually, since it affects, it can prey on other gram-negative. One of the preys is E. coli, and that's one of the most abundant gut bacteria. So yeah, it could cause damage as a means of antibiotic if not modified to target others. I think.
All right.
Yeah.
Thank you.
Thank you.
All right, our next speaker, just the one, just Cole Fulton, coming to us, second year med student as well. Excellent. And you worked with Dr. Breshew in the Department of Physiology.
All right. Good morning, everyone. I'll be talking about step-in-place tests, which is a lot more exciting than it sounds, believe me. The original step-in-place test was designed by Fukuda in the 1950s. He had patients, or subjects rather, step in place with their eyes closed, and he noticed that nobody was really good at staying in place with their eyes closed. He concluded that if anybody had rotated at the end of their stepping test, well, let's see if this goes, there you go. If anybody had rotated over 30 degrees, he called it labyrinthine dysfunction. He also noted that people moved forward, though, and so our interest in this test was to see if there was anything else that we could glean from the movement pattern of these people. We thought it could maybe indicate postural deficiencies, instabilities in their legs, even asymmetries like leg length asymmetries. So we split them into 3 different variables: the total movement distance. We kept the rotational angle, obviously, and the displacement angle, because people could move from the starting position without rotating. So, our purpose in the study was to determine the reliability of the test, see if it was repeatable, and also to identify, like I said, potential anthropometric and functional associations. Right. Let me get that mouse out of the way. We had 5 men and 5 women volunteer. We took measurements like their height, their weight, their leg length, their thigh circumference, and their umbilical height. And they came each 3 days and did 3 trials of each thing on each day. So they did a balance test each day, 3 times on each leg with their eyes closed. And then they did 3 stepping tests each day that were 60 seconds long, blindfolded. This was the position that we asked them to hold for the balance test. If anybody's tried that, it's not easy. We asked them to bring their hip and knee joint to 90 degrees, keep their eyes closed, and then we time them. And when they put their foot down, we stop the timer. This was also the position we asked them to step in, was 90-90, which is also not easy with a blindfold on. And we got very interesting results we'll get to. This was our testing area. We drew a protractor on the floor. We had them start on this back line. One of us sat at this computer and said, go. And this is an infrared optical system that would start the timer as they picked up their foot. And one of us stood on the outside so that as they moved, they wouldn't trip on the equipment or run into it. Like I said, we asked them to step high and relatively quickly, just so we could get enough data points in the 60 seconds. Sometimes we had to put our hands on their shoulders if they got too close to the equipment. And then we had them stop still with the blindfold on, and we recorded their displacement angle and the rotation angle with another protractor we set on the floor. Right. Then we also, well, later we analyzed their total movement distance. As I mentioned before, we triangulated 3 footsteps at a time, 3 points at a time, and took the hypotenuse of each of them and summed them all up. So these are some examples of some stepping tests. So this person, I think it would be good to just kind of demonstrate. This person went straight. Basically, this is the starting position. They were facing this direction, and they didn't have very much of a displacement angle, right? Just 2 to the right, 2 degrees to the right. But they did start rotating, and they ended at 45 degrees, right? So according to Fukuda, this might be abnormal labyrinthine function. Their total movement distance was 285 centimeters. So this was a pretty normal-ish test. This one gets more interesting. This person marched, and keep in mind, these people are trying to stay in place, right? But hooked all the way around to the right and ended up rotating 215 degrees, right? So he ended up looking like this, which was funny to watch, believe me. His total movement distance was 660 centimeters, so getting a little bit longer. This one was very interesting. This person went straight left and at the same time rotated a little bit to the right. Total movement distance was 726, so significantly longer than the first. And the rotation angle, interestingly, was within the 30 degrees that Fukuda said. So this person might have been considered normal to him. But we thought, there's got to be more going on. Right? The last one, this ended up being the longest total movement distance. And as you can tell, total movement distance didn't indicate how far they actually moved. So this is 100 centimeters. So this person only might have moved 70 or 80 centimeters. Well, actually, this person started going to the left and then ended going to the right. But the triangulation between his footsteps, or her footsteps, was such that it ended in a big number for total movement distance. And again, this one was interesting because they went this way and then went that way and also rotated to the right. So as you can see, we had varied results. A little bit of the statistical analysis. The total movement distance was moderately related on day 1 and day 2, but not on day 3. The displacement angle and rotational angle were both moderately related on day 1 and day 2, and only rotational angle stayed related on day 3. The total movement distance was not very related to the displacement angle and rotational angle. Also, unilateral contact time, as measured by our OptiGate system, was correlated with single leg balance time. So if they balanced better on one leg, they ended up spending more time on that leg as they marched. And left and right asymmetry and contact time largely determined the direction of the displacement angle and rotation angle. So they actually moved toward the leg that they spent more time on. So in conclusion, we thought that displacement angle and rotation angle might be related to functional asymmetry. Whether that's strength, could be a leg dominance thing, and we have a colleague that did a whole study on that, so you could talk to him, Trenton. It could be a neuromuscular activation pattern difference in people. Could be postural related. We are unsure what it indicates right now. The displacement angle and rotation angle were also not related to the total movement distance, which was kind of surprising at first, but also the more we thought about it, it made sense that they were measuring different things, we think. We think the total movement distance might have been measuring imbalance because they were unable to pick up their foot and replace it in the same spot. The stepping test reliability overall appeared to be best with the initial trial of each day and the initial trial day. And it just got worse as we tested people because it seemed like they recognized that they were moving, and no matter how much we tried to mask the fact that they were moving, they tried to stop moving. And so we think that affected the reliability. We think actually we might need to wait for more time in between trials, and that might help the reliability. So those are our conclusions. We really had a lot of fun with this test. It was very interesting, and we think it'll give us a lot of good information in the future. That's all I have. Thank you.
It's a very interesting study, and looking at the graphs, whatever you showed, the test subject clearly lost the orientation. So one is going straight, another one left and right. So it clearly shows that apart from the whatever measurement you are doing, that their GPS system in the brain is off, and that's why they are not able to locate their positions they go. So it's more and more clear that their GPS system or the brain plays a major role in these tests rather than any physical thing, because they totally lost their orientation. Like you said, one person turned 180-degree back. So their GPS system is involved. The involvement is more than the other thing. So I think this is a very good model system to study the brain GPS system.
All
It's
right,
good.
yes, it's very interesting.
I was curious what your age range and general fitness level of your subjects were.
We were generally, well, they were generally healthy, and the age range, I think, was 24 plus or minus 2 years. Okay. Our colleagues here.
You mentioned that between day 1, 2, and 3, they did attempt to not move as much, but did the pattern of their movement, were they similar, or were they very dissimilar one day to the next day for the same subject?
Again, they seem to correlate on the first day, not so much on the second day or third day. So the pattern changed. Sometimes there were people that were a little more consistent, but for the most part, the more we tested them, the more it changed.
So you kind of talked about labyrinth dysfunction, and generally the brain is able to integrate vision with the ear modality to see where we're at in space. But when you shut your eyes, it kind of moves into a more proprioceptive focus. So I'm wondering if you guys had them march to like a metronome, that way they all stood on one foot with a similar time, because maybe if they were marching slower, they might have less movement from their original spot.
We didn't use a metronome because, again, it would kind of give them a reference point, and we found that whenever anybody had a reference point, they tended to turn towards it. We also did this in a room with a window, and if people could notice the light, they always turned toward the window, right? So we didn't use a metronome for that reason, but we got it to the point where they were stepping about 1 step with each leg per second, and if they were stepping a little bit less than that, we would just ask them the next trial to speed up, and that's how we kept their pace somewhat consistent. Yeah.
So I was interested in your opening remarks that you said the original study, if someone rotated over 30%, that was identified as dysfunction. Could you elaborate on that and say what was the source? Was there a speculated source of dysfunction? Or do you actually believe, based on this, that there really is dysfunction involved in someone rotating over 30%?
That's so hard to say. I really don't know if I believe that there's dysfunction involved with over 30 degrees movement. I don't know that in the original study there was actually any pathologies. I think there were some deficiencies he noted, like maybe ear infections or something wrong with that side, and they tended to turn toward that side with the defect, but really, I don't know if there is any correlation.
So,
Great question. They'll probably spin in circles, really, stay in the same spot and turn a lot. But really, I haven't read all of the literature. So.
Nice presentation, pretty interesting data. I'm not sure I caught it. Were they barefoot?
Oh, great question. Yes, most of them were barefoot or wearing socks. We didn't regulate that all too much, but
no, I was just
they
concerned
didn't
if they were
have shoes
wearing
on.
shoes, for example, the age of tennis shoes, and whether you pronate or supinate shoes wear out. So I just was curious.
Right, great question. I forgot to mention they weren't wearing shoes.
Very good. Thank you.
Yes.
Thank you for your presentation. I would think that there are a lot of confounding variables here. Great study, great things to look at. But I think age is a fairly large factor here. As not my field, but as you get older, your feet change. And you really can't stand on one leg or one foot very well for some people. So I think as you progress in this, that would be a very interesting thing to look at. Thank you.
There was actually a study published recently by our lab that identified frailty in elderly individuals, and this test was very useful in identifying frailty, so you're right with that, yeah.
Looking at the picture on your lab's setup, it looked like their starting point was pretty close to the back of the square. Which is potentially, I mean, I don't know psychologically whether it would make someone feel like they need to move because they're going to worry about falling back. I wonder what it would look like if you started out 2 feet further into the space. That was all.
That's a good point. In the original test, actually, Fukuda noted that nobody went backward. And I think that's kind of what we went off of, just kind of banked on the fact.
You're not going to go backwards because you know you're not allowed to.
Right, right. Could definitely have an impact. Yeah.
All right, thank you, Cole. All right, our next presentation is by Alicia King. She's an ATSU alum working in Department of OMM at ATSU.
That's going to catch there. Put on this side. Yep. Just trying to keep the stupid thing keeps falling off. I may.
Just forward on this side and backwards. You need to go and there's you push it up on it. There's a laser there.
Awesome. And push there to.
Yep, you should be ready to go.
Okay. So hi, I'm with the OMM department for a final year of residency. And what we were looking at was the characteristics and management of geriatric patients from the neuromusculoskeletal medicine and osteopathic manual manipulation clinic. Dr. Karen Schneider is our mentor and helper with this presentation, as well as one of our fellows, Jamie Cox, and Shalini helped us with the data. So a little bit of background. OMM is very much a recognized complementary health approach. It also has been well known to help in our geriatric treatment patients since Dr. Still's time. We've also had some studies come out in relation to the osteopathic data for treatment in geriatrics, but there's very little that shows what the presenting complaint was, as well as anything that gives why they chose OMT to go along with it, and what treatments they used, the body regions, what modalities they used. So what we're trying to do is get some basic information out there in regards to geriatrics, because this is a very up-and-coming field that we need some more information on. So our goals are to do descriptive data of the geriatric patient population treated at our, or receiving OMM in the neuromusculoskeletal medicine and osteopathic manual manipulation clinic. And our aim is to get the information out to clinicians that are treating these geriatric patients and what OMM seems to be used with them, and potentially even maybe give them suggestions for other things that may also be of use. So our methods, we did a retrospective, descriptive, observational study. We used the electronic health record to obtain the data. We used a single outpatient specialty clinic, and we got our IRB approval through ATSU KCOM's IRB. So, our methods. We had geriatric clinical encounters involving OMM. We looked at a 3-year period from July 1st, 2016 to June 30th of 2019. And the data was categorized by patient's age, and we used that as our basis to select our people and analyze them. We looked at encounter notes, billing records. We looked at a lot of their demographic information, their presenting complaints, the somatic dysfunctions that were assessed, what treatments were used, and what body regions that they were treated. So these are the overall numbers that kind of play in on the next few slides. Our total clinical encounters was 9,155. Our total discrete patients was 1,238. We had a total of 12,020 presenting complaints, and they can actually have up to 5 chief complaints per our EMR. There was 43,862 total OMM techniques used. The way we kind of counted that was for each body region. And then we had the total body regions treated, which was 46,100. And they could have up to 10 body regions. Those of us in OMM practice are pretty used to those regions. So looking at the discrete male to female patient ratio. The numbers aren't quite about 1/3 and 2/3, but we had a little more than 2/3 female population presented in this study, and about not quite 1/3 male population presented. When we split them up by age range, our 60s was our biggest decade that was presented in clinic. Those were 609. 70s were 403. 80s were 190, and we had 36 that were over 90. I think, and if I remember correctly, I think our highest was maybe 106. So. Then we also looked at overall the years just to make sure that there wasn't a huge outlier on any of this. And all this data looks pretty kind of consistent across the board. Our overall was 4.5 clinical encounters of these discrete patients. What numbers in parentheses is the standard deviation for the clinical encounters, and then the N then follows underneath it. Looked like maybe in the first year, maybe there were a few more clinical encounters overall, but not anything that was really standoutish. So when we look at the overall clinical encounters, not just the discrete patients, women typically manage to be about 3/4 of the cases that we're looking at, and men typically were about 1/4 of the cases that we were looking at. So getting down to the meat and bones of what the clinicians want to know. So what did they present with? We had back pain, lower extremity, neck, pretty much everything under all across the board. And then we also had some non-musculoskeletal. And our top 5 were the back, the lower extremity, the neck, the upper extremity, and the hip and pelvis. Just what you'd expect to find in this population. Then we looked at all the OMM techniques that were documented during the clinical encounter. Once again, this is got an N of 43,862. So Yes, your favorite technique is displayed mostly here, and if it's not here, then it's probably under our other. But these were our top 10 OMM techniques. Muscle energy definitely rounds it out at the top. Myofascial release, articular, soft tissue, all the things that should be in the wheelhouse of most of our osteopathic doctors. Steel technique, percussion hammer, indirect counterstrain, HVLA, and also cranial. So what regions did you figure we treated? Well, those are our 10 regions. It's probably not a surprise with the chief complaints that thoracics and lumbar are our top ones, followed then closely by cervical and ribs and pelvis and sacrum. And then further down hits a few of the other places that probably had some corollary issues going on. So what we got out of this, we definitely had a good description of the geriatric patients that came to our OMM clinic. Our most common complaints were those back and lower extremity pain. The most common treated areas with OMM are the lumbar and thoracic. So this is very consistent with some of the meta-analysis data that's come through. The most common sites of pain in geriatric patients, no surprise, it's back and lower extremity pain. And it's very similar to the findings in the surveys of OMM that we're seeing in kind of across the literature. The most common OMM technique used is typically muscle energy. We found that in our study. We've seen that in our osteopathic physicians in practice, and we also do see that in our students taking the COMLEX. The weaknesses of the study. It was done off of EHR data, so anything that was more written in was excluded from the data, and that may skew that. We also looked at a single clinic, so this would not be representative to all the clinics. It would be something to look at, but at this point we don't have that information. We also did this as the OMM NMM clinic, so it wouldn't always be translatable to the primary practice, and that would be another place that we could look at. So there's definitely consistency in the OMM. It's current with in our training of our osteopathic medical students, and it's also very, very consistent on the practicing osteopathic physicians in clinical practice. It's a section of high-value research, mainly because we've also had several recent studies that have shown OMM as an effective treatment for geriatric musculoskeletal pain, and it's also in the current treatment for back pain. So what we have concluded is that our data can be used by the clinicians to at least identify the role of OMM as a geriatric complementary health approach, and it also identifies other potential conditions for future outcome studies. And then, once again, like I alluded to, our future research would be to look at other locations, look at if there is any variation in the presenting complaints, techniques or areas, as well as to compare nursing home patients to the regular population, because we have some nursing home patients that do get transported over for our OMM clinic, but we don't have a huge population, and that wasn't specifically delineated in our study. Any questions?
Okay, so from the statistics, whatever you present, it shows you have more, the female number is more. Mm-hmm. So whether it's due to whether they complain more or you have access to the female, more number of female data. Why the female number is high? Because they often complain, or what is the reason?
Well, in a lot of medical studies, we do find out that females typically do complain about more of it. There's also a demographic bias in general. Females typically live longer than men. So whether we're showing that as kind of what the demographics of the area is, that would be a good thing to look at.
And in the geriatric, in the older patients, the chief complaint is on the weight-bearing area, like back as well as lower extremity. And then if you look at the abdominal complaint, it's just around 700 compared to the other which runs more than 2,000. And the chief main complaint is the weight-bearing area. So is because of bone structure, or what could be the reason?
Well, it can be anything from, and we didn't look at a lot of that, would be like, these are also going to be the patients' populations that are going to have a lot more arthritis, they're going to have a lot more osteoporosis, all those wonderful diseases that we kind of accumulate as we grow older, they hit this population, honestly, probably the hardest.
Did you do any differentiation in gender for the various complaints, particularly your top 10 complaints?
I did not. That'll be something that we look at for future.
Alicia, impressive number of subjects and a lot of work. I guess, I don't want this to sound wrong, but just because something is done doesn't necessarily make it right. Have you looked at the outcomes and the efficacy of the different treatments in your subjects as the next step, and saying, what we did this, and just because we did it work, and then actually assess that outcome? Have you done any of that?
We didn't. One thing that I kind of does allude to some of that, though, is that it was the amount of clinical encounters per year is about 4. 4.5, I think, is our overall. That would suggest that maybe they were getting better within that time frame. That, or we didn't do any good. Somewhere along the line, at least, typically that's about where they fell off.
It would be really nice to follow this up with any indication of the outcome and really look for the efficacy of different treatments.
Definitely. Thanks for the.
So I was interested in one of your limitations being a single clinic, which made me think, are there regional differences in techniques that you're aware of, so that you would actually predict that the data could be different, whether maybe not the chief complaint, but the actual methods that were used to address these complaints?
Yes and no. A lot of our different, where some of the specialty clinics are located, a lot of those also are very strong in muscle energy as well. So I'm not sure that would really change that aspect of it, but it may delineate some of the other treatment modalities.
Dr. Sexton alluded to the potential power of the electronic record. You eventually see the patients again. Is there any place in the current electronic records to get that outcome? Ask the patient if their penicillin helped them get over strep, or their OMM helped them? Is that in the record at all?
Well, at the end of every treatment, we have to put whether the patient improved, didn't improve, whether it was resolved. I'm not sure if we've specifically coded to look for that, and that would be something else to look at. But long-term, short of specifically asking the patients individually, our EHR doesn't really have a spot for that.
All right. That'll be it for now. Thank you.
Thank you.
You can always see her at her poster. All right, our last speaker, Clara Wolf, a Truman State student, I believe, that also did some work at WashU. All right. So while she's getting set up, after she is done, we'll have about a 15 to 20 minute break, but we would ask that you come back in here at about 5 till, so that we're going to be showing a short little video and then get into the keynote address by Dr. Magruder, which is strange to say, Dr. Magruder, and it's a different one, right? It's great to have you here, and we hope that you guys stick around for that.
Is the middle the PowerPoint? Is the middle the laser pointer? All right. All good. All right. Okay. Oh, okay. Thank you. Good morning. My name is Clara Wolf, and today I'm going to be telling you all about the research I did this summer at WashU's Mallinckrodt Institute of Radiology within Dr. Tammy Benzinger's lab. And so this summer, I studied the relationship between physical frailty, brain atrophy, and cognition across adulthood. So what is physical frailty? Physical frailty is a measure of physiologic risk for developing certain conditions and or risk for developing cognitive decline in the future, as well as certain post-surgical outcomes. The reason we were interested in physical frailty is because previous studies have shown that physical frailty can be a measure of physiologic resilience toward developing certain diseases or certain symptoms associated with diseases such as Alzheimer's. So one study showed that individuals who had the same pathologic burden of Alzheimer's would be more likely to develop cognitive decline based on their frailty status. So those who are more frail were more likely to develop cognitive decline sooner than those who are non-frail. Furthermore, a lot of the studies that we had looked at previously only studied those older, 50 or older or 60 or older, and so we wanted to look at physical frailty as a measure across adulthood and not just in older populations. So we predicted that more physical frailty would lead to more atrophy in the brain, or would be associated with more atrophy in the brain, and this atrophy would be associated with lower cognition or lower cognitive scores. So we thought that physical frailty was affecting cognition through this mediating analysis of, or mediating relationship with brain atrophy. And to go about testing our predictive model, we had 87 participants for which we collected data. Our age range was 31 to 91. As I mentioned, we were looking across adulthood. And our participants underwent 3 assessments. One was the Johns Hopkins Physical Frailty Assessment for physical frailty data, as well as the Montreal Cognitive Assessment for cognitive data, and an MRI. So what is the Johns Hopkins Frailty Assessment? As the name mentions, it's a frailty assessment, but it's a composite frailty index, and it uses 5 criteria. The criteria is scored 0 or 1, 0 being normal, 1 being abnormal. And so each criteria is given one of these scores, and these scores are summed. Sums of 0 are given a frailty status of non-frail, and 1 to 2 is pre-frail. 3 or more is frail. And we did not have many participants determined to be frail as their frailty status. And so for our analysis, we grouped pre-frail and frail individuals. One thing to note is that our biggest predictor of our 5 criteria of frailty status was grip strength, measured in kilograms. And so we decided to use grip strength as a separate measure of physical frailty, not just physical frailty status. So in doing our analysis, we had 2 measures of physical frailty status, and grip strength. Furthermore, for our cognitive data, we had data for a subset of the participants of 71. So we used 12 regions of interest, or ROIs, to test our atrophy data, or as our atrophy data. 10 of these regions were cortical thicknesses, and 2 were subcortical volumes. All had been studied previously and related to Alzheimer's previously in literature. And we used the T1 MRIs, and we used the program FreeSurfer to segment the different regions of the brain and to give us the volumes and the thicknesses of our particular regions of interest. So then, in using all this data to test our predictive model, we used linear regression in R, and we controlled for age and sex in all of our tests, or all of our analyses, as well as education with our cognitive data. And this is the results, the background characteristics of our participants. As you can see, and probably would have guessed, those who are pre-frail or frail are significantly older than those who tested to be non-frail. Furthermore, the MoCA, or the cognitive data, seems to be trending towards having lower scores in those who are pre-frail and non-frail, but this was not, we didn't find that to be significant. Finally, you'll see that grip strength is very significantly lower in those who are pre-frail or frail, but as mentioned, grip strength is one of the indicators of whether an individual is pre-frail or non-frail, so this makes sense. So we started testing our predictive model, and first we wanted to see if physical frailty was predictive of brain atrophy. And as I mentioned earlier, we had two tests to tell us two measures of physical frailty, the physical frailty status and the grip strength. And we found that, oh, that got moved a bit, but we found that physical frailty status was not predictive of atrophy in any of our 12 regions of interest, but we did find that grip strength was predictive of atrophy in 6 regions. Also to note, the MTL region is a medial temporal lobe region made of 4 composite regions, and we found significance within that region. We tested the regions that made up that composite region. I apologize, the box got shifted a bit, but the ones in red are the regions we found significance with. So these are the graphs of those 6 regions. And on the x-axis, we have average grip strength, and the y is the thickness or the volume of each region. As you can see, the relationship includes that lower grip strength is associated with lower or thinner or lower thickness and lower volume in the regions. The most robust relationship comes with the hippocampus. And so from here, we wanted to see whether these 2 measures of physical frailty were predictive of cognition. And we found that neither frailty status nor grip strength was associated with our cognitive scores or our cognitive data. And so lastly, we wanted to test if atrophy was associated with cognition. And we found 2 regions for which the volume or thickness was associated with our cognitive data. These 2 regions are the hippocampal volume and the parahippocampal thickness. Similarly to the last graphs, there are positive correlation with the lower hippocampal volume or lower parahippocampal thickness associated with a lower MoCA score. And although we found that grip strength or lower grip strength, so more physical frailty in that assessment, was associated with more atrophy in the hippocampus, and more atrophy in the hippocampus was associated with lower cognitive scores. Because we did not find that physical frailty was predictive of cognition, we cannot say that this supported our prediction of the mediating analysis. So our results did not support our hypothesis. We did find it interesting, however, that grip strength was a better predictor of brain atrophy than the composite frailty index. So this leads us to maybe suspect that measures of physical or muscular strength might be better predictors of atrophy than a composite, or brain atrophy than a composite frailty index, potentially. Furthermore, as I mentioned, we did not find any association with physical frailty and cognition, but one of the biggest limitations with our study is that we used the Montreal Cognitive Assessment, which is a good test for global cognition, but there are definitely more comprehensive, more robust cognitive assessments that exist. And so in the future, we hope to score some more of that cognitive data, because especially given we have a larger age range of participants, this data might be more sensitive to differences within our participants, potentially. And that's a part of our future direction, as well as to look at different cognitive assessments that test for different cognitive domains, particularly looking at cognitive domains that are associated with the regions of the brain that we found atrophy in might be interesting. We're also looking at other measures associated with frailty and physiologic resilience, such as cerebral hypometabolism in the future, as well as how different life choices or different lifestyle choices in young adulthood lead to resilience, physiologic resilience in adulthood. And I'd like to thank, oh, that got messed up too. I'd like to thank everyone in the Benzinger lab and the Vlassenko-Goyle lab and everyone at WashU's Neuroimaging Lab for allowing me this opportunity, as well as the Amgen Foundation. And these are my references, and any questions?
I'm gonna be a bully and take one question. They used to, before GPS came along, they did a study on taxi cab drivers in New York and saw that their hippocampus was monstrous compared to general population.
Mm-hmm.
Thinking of that, because they just memorized all the streets, right? Now they don't do that. But thinking about your subject population, was there anything in their background demographics, their work life, that would lead you to conclude any particular differences that they started with in brain thicknesses?
I don't believe so. I think when we looked at our background characteristics, nothing particularly stood out, but that'd be interesting to maybe add occupation to as one of the background characteristics.
Yeah, I have a question. Like, this is interesting, study. But the sample is pretty much like small. We have like 78 patients, and only 2 patients classified as frail.
Yes.
So do you have any like software to calculate like how much the sample, the sample size to go, with the good conclusion? Because that's
we are actually working on repopulating the study to repeat it. So, because we think the best way to test rigorously this result is to repeat find the same results in a very similar population. So we hope that just by finding this again in a separate group of participants, that we think that would be more rigorous than doing something like a multiple comparison correction. So that's our future studies.
So cortical atrophy has been shown to be associated with chronic pain. Did you collect pain data on these people?
Oh, we did not.
It seems to me that time is really sort of your enemy here, and if there's a temporal offset in sort of when frailty develops and when its consequences would show up or something like that, you get in trouble. Do you have the ability to do sort of a case control kind of design where you start with just a whole lot of frail patients and a whole lot of not frail patients and look backwards at their, somehow look backwards at their index of grip strength or something, doing the study the other way around in a case control kind of design?
I do not know, but I do know that all the participants in this study, I think, are going to be coming back, because the participants in this study are also participants in other studies as well. So I think that all these participants will be coming back for the next, I think, 3 years to repeat the same tests, as well as other tests in these particular studies. That's from my understanding, but that'd be very interesting to look at to see the sort of like a longitudinal study, I guess.
So your grip strength correlation with atrophy, brain atrophy, did you look at the source, and did any of your MRI data include motor cortex to see if there was a correlated atrophy in motor cortex with the reduced grip strength?
So we did not actually look at the motor cortex, but I think that would be another region to potentially look at. But a lot of the regions we found, or at least some of them we wanted to look at, were associated with sort of like early symptoms of Alzheimer's. But that would be an interesting reason to add to our regions of interest.
Okay. The same question I thought of asking, how did you correlate the brain atrophy with the MRI data or CT data? So which one you use to correlate the grip strength with the brain atrophy? So which test you use, MRI or a CT scan or?
MRI.
MRI. And the other question is, the hippocampal area is involved in emotion. So did you study their emotional intelligence in these people or emotional quotient in these people?
I did not.
Okay.
So in your graph, your hypothesis is that physical frailty is affecting hippocampal volume and cognition. And it kind of strikes me that arrow may go both ways. Is there any studies that suggest that there is an effect the other direction, that cognition may be affecting your brain volumes, and that may be affecting frailty, that they're sort of interacting, play between the two of them?
For sure, we definitely need more studies to conclude that, but for sure it's just sort of an association, not very predictive. The arrow definitely should go both ways for that, but I guess sort of looking just at the associations between the two, and then from there we maybe do more research to conclude whether which one is predicting the other, or is there that association exists, but that'd be for the future work.
All right, thank you very much. So we'll take a short break, and we just ask that you be back in here at about 5 minutes till 11 o'clock. Thank you.
Okay, it's time for us to begin getting ready for our keynote presenter. And it's also a time for us to recall a very important person that was critical in the establishment of this research symposium. For 8 years, Neil Sargentini and I worked together in establishing this conference, this symposium. And I can say without a doubt, I have never experienced somebody who was so selfless and tireless in promoting education for his students, promoting research and scholarly activity as well. And as a result of that, we have created this memorial lecture in his honor, and I'd like to just offer you these recollections of who he was and his attributes to KCOM.
I just wanted to say a few words about Dr. Sargentini as a researcher. He earned his PhD in medical microbiology from Stanford University in 1980. His dissertation topic was all the genes that control DNA repair in Escherichia coli, especially those responding to radiation damage. And in fact, he was curator of the Stanford radiobiology collection that included 4,000 different strains of Escherichia coli.
I had the distinct pleasure of working in Dr. Sargentini's lab for 8 years, and when he first hired me to work in his lab, he was just simply my boss. And then he quickly became my mentor because he had to teach me all the methodologies and how to conduct his research. And then eventually he started teaching me as well how to mentor his biomed students and other students that would come in to do research in his lab. And then about 3 years ago, he became a collaborator with me, and he allowed me to start doing my own research, which is not heard of very often, to allow someone who's supposed to be doing solely the investigators, the PI's research, to allow them to branch off on something completely unrelated. I will forever be indebted for him for allowing me to do that.
I'd like to talk to you about Neil Sargentini as an educational leader. When he first started as chair of our department was doing okay, but they weren't doing great with regard to educating our medical students. Dr. Sargentini, not only was he interested in having us become an educational leader in our college, but he was competitive. He wanted our department to do better. And because he wanted us to do better, he had a vision for improving. And his vision really resulted in us becoming a good. We went from being an okay department to being an excellent educational department for our students.
Dr. Sargentini, I truly appreciate for being a good example to me and many others. You showed me how to be a good racquetball player, a good teacher, a researcher, and above all, a good human being.
You know what I saw that I really liked about Neil was that he sought to master the things that he got involved with. Didn't always know what he was getting into initially, but he did his research, he was due diligent, he got on top of things, and then he had a well-organized and systematic approach to getting things done. That really lent itself to when he came over to play racquetball one day, and he didn't move the best for, you know, even, I'm not sure about his athletic past, but I think it was probably mostly PhD work. So as Neil played a little racquetball, he really learned about the dimension of the court and the angles and how to minimize his moves and cut the ball off, and kind of became better at playing this game and played with several friends a couple times a week. And so what he started to do is say, Dan, I think I can organize these 15 people playing in a league. And I'm thinking, gosh, maybe Neil, I'm not doing that well at it. And he helped us. And as Neil got more involved and held beginning clinics and was so embracing and wonderful with the students who found him to be nurturing and insightful, we had 75 and 80 students playing in the leagues by the end. For 15 or 20 years, he did this with us 3 or 4 times a year. And so we got to know Neil pretty good. I remember when Vineet Singh came to campus, and Vineet had played some squash in the past. Of course, he's in microbiology, and he went in there, and Dr. Sargentini just waylaid him, just beat him terribly. Dr. Singh also likes to master things. So Dr. Singh worked as hard as he could to get better than Dr. Sargentini, and he got in better shape.
I remember those days when you got me interested in the game of racquetball. I remember those days when you gave me 5 service chances and I still failed to score. But I had a desire to be really good in that game, and with your coaching, I soon became even better than you.
Something that was really remarkable about his career was the fact that he was able to include so many students in his research laboratory, actively doing research that was very publishable and that really benefited the students as far as their educational experience.
Dr. Sargentini was a splitter, not a lumper, and he didn't want to forget anything, and so oftentimes in his thought was it was better to include too many organisms than not enough. And so we went through and figured out where our gaps were and worked hard on making our department a better department at educating. And because of him, we are better.
And what I really admired about Dr. Sargentini was that he was always open to change, and he was always open to improvement, and he never was stagnant, and that is a rare gift. So over the course of 8 years, he was my boss, and then he was my boss and my mentor, and boss, mentor, collaborator, to eventually just one of my favorite people.
And I've learned so much from you. I am so proud to have worked with you, learned with you, and I will always be grateful for everything and all of those things. You will always be in my memory. Thank you, Dr. Sargentini.
So I don't think there's any more fitting way to honor him with this memorial keynote lecture. And this year, it is my honor, my privilege to introduce to you Dr. Carrie Magruder. Carrie is the curator of the History of Science collections at University of Oklahoma Libraries. He is the John H. and Drusa Cable Endowed Chair of the History of Science and is the Associate Professor within the History of Science Department at the University of Oklahoma. He is co-editor of the peer-reviewed scholarly publishing series called Edition Open Sources, in collaboration with the Max Planck Institute in Berlin, Germany. He has lectured extensively throughout the United States and has numerous publications within his field. His roots are here in Kirksville. He was born in the Truman State University Police Department, which was at that time actually the Grim-Smith Hospital. He went to Northeast State University, graduated with his degree in biology, and at that time had a decision to make. He was very strongly interested in the field of biochemistry, but knew that he wanted people to really understand and appreciate what science was. And that's what drew him into his master's degree in education, where he got his master's at Truman in science education, and then brought those skills to St. Louis University, the city of St. Louis, where he taught there for several years, before he decided to go into his PhD program, where he received his PhD in the history of science there at Oklahoma University. For him, science is really important as a story. And I would say for us as well, even though we all use science in our own disciplines and our own areas, science is the story that brings us together today. It is an area that certainly we have in high esteem for establishing directions and priorities for how we care for humankind within the biomedical sciences. It is something that at various eras have come into question, and it certainly can be criticized and for how it's used, and certainly can be sometimes ignored or even undermined as a way of trying to sustain ignorance. Today, Dr. Magruder is going to be paralleling us two of these times, and how a person in previous time in science history, who was often considered the father of physics, the father of science, the developer of kinematics and in other areas of science, how that person's experience, that person's culture is still relevant today and in the future of health sciences research. So please help me in welcoming Dr. Carrie Magruder.
Thank you, Dr. Degenhardt, for that wonderful introduction and for inviting me to be with you today. It is humbling to be here and to honor Dr. Sargentini. It is a true joy to be able to spend the whole day with you, and I feel an intrinsic joy welling up within me as I have experienced the window into the work each of you carries out day by day that's presented through the research that we're celebrating today. So it's very exciting to me. It's a privilege to be here at this historic institution where each one of you knows the value of the stories of science that Dr. Degenhardt alluded to as part of what shapes our professional identity. There is a research side that is an essential component of professional identity, but here with the museum and the historic tradition of A.T. Still University, you already know that what happened 100 years ago is part of the fabric of what creates your day-to-day life here. So that's what we're going to do now. In fact, this would be cheesy, except the former president is going to do this with us. Please remember Dr. Still and his techniques. Put some fingers on your temple. If everyone can do this, see? Yeah. All right. Repeat after me. I will enter the professional identity zone, ready time machine, T minus 400 years, engage. All right. Let's see if there are stories from forgotten stories from a bunch of old books that might be relevant to what you do here. So, and as an aside, at various times I'll refer to a Galileo exhibit we did back in 2015. My thanks to Susan Swanker and Deb Laguda Summers for putting some of the graphic panels from that exhibit on display here to celebrate this very symposium. To dive deeper into that, Links Open Ed is the website for educational engagement that I and a graduate student have created. You'll find an exhibit guide there of more than 1,000 pages if you pull down on the courses and exhibits. And over the coming week, I'll post a PDF of the slides for this presentation at that location as well. And as one other aside, research universities are often best known for their athletic programs. But among historians of science around the world, the University of Oklahoma is known for the history of science collections. We hold about 100,000 volumes preserved in 2 climate-controlled vaults. All images of rare books used this morning are in the public domain from the history of science collections. We have 11 faculty members in an academic department. We offer both graduate and undergraduate degrees in the history of science, technology, and medicine. None of you need any extra degrees, but perhaps it's helpful to know that a program like this exists. And I hope that you will, during your career here, find occasion to do research in the A.T. Still Museum and in the library archives. But we also offer short-term travel fellowships for research projects in the history of medicine. But with that done, let's address our theme today. And first, we'll take a look at connections. Then we'll explore innovation in Galileo's world in 3 areas: instruments, medicine, and anatomy. And finally, we'll conclude with some reflections on innovation in Galileo's world in terms of synergy and exaptation. Synergy means the whole is more than the sum of the parts, and exaptation refers to the productive transfer of skills or knowledge from one domain to another. So let's begin with Galileo and connections. Galileo is regarded as a founder of modern science because of his contributions to astronomy, mathematical physics, and experimental methodology. This is the OU copy of the Starry Messenger. It was inscribed by Galileo in the lower right corner. In the Starry Messenger, Galileo printed the first published observations made with the telescope. He reported mountains on the moon at a time when physicists in the universities believed that the lunar surface was smooth. His discovery of the 4 largest satellites of Jupiter demonstrated that multiple centers of revolution must exist in the solar system. That proved that a moving planet need not outrun a moon. Unsuspected stars in the Pleiades, around Orion's belt and sword, and in the lighter regions of the Milky Way suggested the vastness of the universe. These discoveries made implausible the ancient notion of a celestial sphere carrying fixed stars and rotating around the Earth every 24 hours. The sensational discoveries revealed by the telescope catapulted Galileo onto the world stage, making him a celebrity almost overnight, and to this day. Galileo's letters on sunspots showed that the sun is not changeless either, which suggested that the heavens are corruptible, contrary to Aristotle. Galileo reported that through the telescope, Venus shows a complete set of phases, which implies that it revolves around the sun instead of around the Earth. Although the phases of Venus were compatible with the Earth-centered cosmologies of Marcianus Capella, Tycho Brahe, Riccioli, and others, Galileo interpreted all of these discoveries as supporting the sun-centered cosmology that Nicholas Copernicus published the previous century in On the Revolutions of the Celestial Spheres. This much is well known. Still fairly well known is that Galileo published his masterwork in mathematical and experimental physics, The Discourse on Two New Sciences, in 1638. In this book, he laid a foundation for mechanics, a new science of motion. Drawing on the impetus tradition of Jean Buridan, Galileo proved that projectiles follow a parabolic trajectory. Galileo also demonstrated that falling bodies accelerate so that the distance they traverse is proportional to the squares of their times, regardless of weight. John Philoponus, working in Athens in the 6th century, dropped objects from a tall height to show that they do not fall at speeds proportional to their weight. That experiment attributed to Galileo is one he never claimed to do at the Leaning Tower of Pisa or anywhere else, nor did any eyewitnesses claim that he did so. Philoponus' work was well known. Rather, Galileo's insight was to use an inclined plane to slow the rate of freely falling bodies to a speed that could be measured. The inclined plane experiment, rather than the Tower of Pisa, best represents Galileo's new physics. So how did Galileo create a new mathematical physics? In an era when physicists were not trained in mathematics but sought logical demonstrations based on qualitative principles, Galileo pioneered an approach that instead combined mathematics and experimentation. So this much is fairly well known. What is not very well known is that Galileo was a physician. That's part of it, but what I really want to emphasize is that Galileo connected the entire circle of subject areas, some of which are shown here. Few people appreciate, for example, Galileo's involvement with medicine, our topic for today. Galileo's scientific innovations depended upon a culture of interdisciplinary connections. In typical Renaissance fashion, Galileo was a polymath. Instead of celebrating Galileo as a founder of modern science, let's shift our attention from Galileo himself to attend more carefully to the culture that surrounded and nurtured him. That interdisciplinary culture made Galileo's innovative work possible. Connections occur in two dimensions. First, connections between the disciplines, as shown in the circle of subject areas. How did the culture of connections between disciplines back then create conditions for creative innovation? And second, connections between Galileo's world 400 years ago and our world today. How might we recreate a similar culture of interdisciplinary innovation in our universities and research groups? So to get an idea of this culture of interdisciplinary connections, let's take a brief look at two examples involving the fine arts. First, some connections between art and science in Galileo's world. In the exhibit, we inquired, what was it like to be an astronomer in an era when art and mathematics were intertwined? For this gallery, my introduction presented some bold theses. In The Starry Messenger, Galileo reported his discovery of 4 satellites of Jupiter and mountains on the moon. These sensational telescopic discoveries were made possible by Galileo's training and experience in Renaissance art. Galileo's scientific discoveries occurred in the context of a specific artistic culture which possessed sophisticated mathematical techniques for drawing with linear perspective and handling light and shadow. When Galileo peered through his telescope and discovered mountains on the moon, he did so because he was seeing with the eyes of an artist. Contemporaries without artistic training were not able to see what Galileo saw. They were able to look even through his telescope, but not to see. Because telescopes at that time were of such poor quality, Galileo's discoveries were not made because of the optics, but it was his eye that made the difference. As a young man, Galileo was trained in perspective drawing. He worked his way through this manual by Sirigati, recreating each of its 65 plates of drawings. Imagine on this one that each spike is the same lunar mountain observed at different times under different angles of light. Careful study and redrawing of the spikes on this ring and the shadows they cast prepared Galileo's eyes before he ever picked up the telescope to be able to interpret the shadows cast on the moon by mountains and other topographical features. So the revolutionary astronomical discoveries of Galileo grew out of modes of inquiry and practice initially developed in art for perspective drawing. The appropriation of linear perspective drawing from art to astronomy is an example of innovation by exaptation, the transfer of a new skill or knowledge to a new domain. An artistic sequence of linear perspective drawing from Leonardo to Sirigati to Galileo, that's the cultural context for the story of Galileo's telescopic discoveries. For a second example, again from the fine arts, let's look at connections between music and science in Galileo's world. And for this, we'll turn to Kepler. And a most astonishing example of music guiding astronomical inquiry is Kepler's Harmony of the Universe, which contains his laws of the motions of the heavens, including the first appearance of his harmonic law. It's written in musical notation. In this work, Kepler was thinking musically. He regarded this work primarily as a contribution to music theory, more so than astronomy. Kepler achieved a synthesis of his new astronomy with recent polyphonic music theory. Kepler demonstrated that the motions of the planets consist of precisely the same harmonic ratios as the latest tuning of the musical scales. The mathematical beauty of music provided the cultural context for what we know as Kepler's third law. Jonathan Annis, an OU graduate student in music composition, composed a suite for harp, flute, and oboe entirely based upon the musical themes from this work of Kepler's. Jonathan arranged the themes, but they're all from Kepler's musical description of the universe as a cosmic dance. Here's a 15-second clip. The whole thing's about 15 minutes. It's great listening under a starry sky. But that musical mode of thinking of Kepler that led him to formulate that law still governs the motions of planets, stars, galaxies revolving around other galaxies. This transfer from music theory to astronomy is a stunning example of innovation by exaptation. So a whole circle of different subject areas, some of which are symbolized here, reflect that interconnectedness of science and culture, which characterized the world of Galileo. But how does Galileo's world connect to our own day? Let's glance at three more quick stories of connections that moved beyond Galileo's narrow world nearly 400 years ago in Tuscany to larger and wider worlds. First, consider this book, bound and cased in a typical Asian style. We've all heard of Shrek, but have you heard of Johann Schreck? Johann Schreck was a friend of Galileo's who assisted him during his initial telescopic discoveries and then went with Galileo to Rome to show the telescope there. Soon afterward, Schreck went to China, where he wrote this work on engineering in Chinese with Chinese collaborators. How many of our Asian students know Galileo had a friend in China? Here's a second example. This book by Francisco Hernandez is the most important early natural history of the Americas. Hernandez spent 7 years with the Aztecs in central Mexico. Galileo and his colleagues in the Academy of the Links worked to publish a definitive edition, which finally appeared in print nearly 80 years later. The scientific and medical knowledge of Native Americans transformed European science in the age of Galileo, and Galileo had a role in the preparation of this work. So the story of Hernandez brings together the Americas and the world of Galileo. How many of our Native American and Hispanic students appreciate that the life sciences and medicine of indigenous Americans was so important to Galileo and his generation? Consider a third story. Johann Hevelius was the leading European telescopic observer in the mid-17th century, one generation after Galileo. This massive book was the first comprehensive lunar atlas. It accurately mapped the moon within 40 years of Galileo's telescopic discoveries. On the frontispiece, Hevelius portrays Ibn al-Haytham, a leading medieval Islamic astronomer and optical theorist. But on the right, holding a telescope, is Galileo. Who would have guessed that one of the most important works of the scientific revolution would portray Galileo in Middle Eastern dress as a tribute to Islamic science? So we'll have more to say about Hernandez in a few minutes, but for now, we've seen Galileo's world connected Asia, America, and the Middle East. And if Galileo's world brought worlds together in the past, are there lessons in this that we may learn from such connections that apply to ATSU and to our research groups? So that's an introduction to the culture of interdisciplinary connections in the world of Galileo. Now let's turn to Galileo's innovations and instruments. In this section, we'll look at the stories of three of Galileo's instruments: the compass, the thermoscope, and microscope. Galileo was an engineer in the Republic of Venice. Venetian engineering was the cultural context for his innovative work with instruments. We'll begin this section by looking at a few examples of what it meant to be an engineer. Galileo began grinding his own lenses as early as 1609. He continued to prepare lenses for telescopes and microscopes until late in life, both in Venice and then later in Florence. Galileo designed this lens grinding machine when he was 75 years old. The book on the left displays a machine designed by Ramelli, a generation before Galileo, which employs the Archimedean screw mechanism for transporting water. Galileo gave judgments to the Venetian Senate on the viability of complex machines like this. Schreck took a copy of Ramelli with him to China, and on the right is the same machine from the book that Schreck wrote after his arrival in Beijing. Here's a book on fortifications by Lurini. Drawing upon Archimedes, Lurini asserted that all machines could be reduced to the balance and thus to the lever. From his home in the Republic of Venice, Galileo taught a private course on fortifications. Both Lurini and Galileo were shaped by a common Florentine tradition in engineering. Tartaglia's compass, it's also known as a sector, incorporated the functions of a quadrant and a caliper measuring device. Tartaglia's new science investigated the ballistics of cannonballs, laying a foundation for Galileo's studies of projectile motion and free fall. Tartaglia taught a teacher of Galileo. The Museo Galileo in Florence holds Galileo's sector compass. From a distance, it looks like Tartaglia's, but it contains innovative scales of Galileo's own design, which are an ancestor to the slide rule. Galileo's first printed book was a manual for his sector compass. The scales were depicted in a later edition. Galileo provided the instrument and the manual to students who were boarding with him for those engineering tutorials. The second instrument is Galileo's thermoscope. For the tradition of pneumatic engineering, let's go back to the 1st century when Hero of Alexandria fashioned all sorts of marvelous machines using steam, air pressure, hydraulics, and falling weights. Once the altar portrayed in this image is lighted, the temple doors open automatically. Just imagine that occurring. Wouldn't that be freaky? Hero's inventions were an important source for Renaissance engineers like Galileo. Galileo's thermoscope, developed in the tradition of Hero, was an ancestor to the thermometer. If a glass tube is filled with water, then inverted and its lower end placed in a small decanter, the water level will rise when heat is applied, say by holding the decanter in your hand, or fall when the room grows colder. The thermoscope relies upon the principle that the volume of water changes when the temperature changes, expanding at higher temperatures and contracting when it is cooler. Galileo pioneered scientific investigations with the thermoscope, along with his two Venetian friends, Santorio and Giovanni Sagredo. Together, they quickly improved the thermoscope in two ways. First, by adding measuring units, and second, by sealing the tube at both ends. Until the glass tubes were sealed at both ends, early thermoscopes measured the mixed effect of air temperature and atmospheric pressure. So the development of the thermometer and barometer occurred together as a result of this thermoscope. Santorio affixed measuring units to the glass tube, which marks the transition from the thermoscope to the thermometer. Sagredo attempted to standardize the scale to correspond to the temperatures of snow, of salt water on a cold day, and of the hottest day of summer. We're a century before Celsius and others. Well water does seem warmer in the winter once our senses grow accustomed to the cold. And so there was a common belief that well water is warmer in the winter than in the summer. But with the thermometer, they were able to demonstrate that well water is actually colder in the winter. Our senses deceive us. The instruments open up new phenomena. Now, in the wake of Galileo, Grand Duke Ferdinand II of Florence established the Academy of Experiment, which carried further the research program of Galileo and his friends. The Academy of Experiment pioneered meteorological investigation using the thermometer, barometer, and air pump, and also medical investigation. The third instrument we'll single out is Galileo's microscope. Galileo devised a tube containing 3 lenses, which he described in this book as a telescope accommodated for viewing the very small. A friend, Johann Faber, named it the microscope. Galileo wrote his friend and patron, Prince Federico Chesi, "I have contemplated a great many animals with infinite admiration. Among them the flea, most horrible. The mosquito and the moth are beautiful. And with great satisfaction, I have seen how flies and other tiny creatures can walk attached to mirrors and even upside down." Chesi and Francesco Stelluti used Galileo's microscope to study the bee. They published the Apiarium, the first published report of observations made with a microscope. And if you haven't heard of it before, that may be because the OU copy is one of only 4 extant printed copies. Another work they wrote at the same time is Stelluti's critical edition of the poetry of Persius, an obscure Roman poet. Stelluti added scholarly annotations on any topic that interested him that Persius happened to mention, including a discussion of bees. So in this plate, Stelluti depicted structures of the bee as they appeared under Galileo's microscope and as described in the Apiarium. You can learn more about the Apiarium and download this English translation from our educational website, Links Open Ed. It deserves to be more widely known. It's very entertaining to read. So in this section, we have seen a circle of subject areas interconnecting in the creative fashioning of innovative instruments by Galileo and his friends. The interaction and transfer of skills and knowledge between these domains reflect a culture that promoted interdisciplinary synergy and acceptation. So that's instruments. Now let's turn to Galileo's innovations in medicine. And here I'd like to return to the story of the Lynx edition of the Hernandez, the most important early natural history of the Americas. The biological investigations in which Galileo participated were carried out by the Academy of the Lynx. The Lynx joined together many of the leading naturalists and physicians of the day. In antiquity, the lynx was renowned for possessing sharp eyesight, even at night. The founder of the Academy of the Lynx, Federico Chesi, believed that the eyes of the lynx, the Academy, would peer more deeply into the secrets of nature than ever before. The keen eyes of the Academy of the Lynx stretched the boundaries of European thought. In the life sciences, with the microscope and the Hernandez, with the telescope. Galileo proudly included the emblem of the lynx on the title pages and frontispieces of his subsequent books, and added the title Linceo, meaning member of the lynx, after his name. Stelluti's edition of Perseus displays the emblem of the Academy of the Lynx, just as he added a scholarly annotation on the lynx, as he had with the bees. This early book by Colonna, a member of the Lynx and an editor of the Hernandez, is the earliest book to contain copperplate engravings of plants. The 26 engravings show much more detail than was possible with woodcuts, as is evident by comparing Colonna's woodblock borders with the engravings of the plants themselves. This plant, fu, was believed to be a cure for epilepsy, and that was a central motivation of Colonna's studies. Let's consider a few examples in the Western tradition that would have been familiar to naturalists. This is the earliest published work on agriculture, a manual for managing a feudal estate. It is an ancestor to the early printed herbals. It explains what plants one must cultivate to be able to make the common herbal remedies. Leonard Fuchs, a professor of medicine at Basel, Switzerland, extracted the best knowledge available from ancient and current sources. This book contains descriptions of 500 plants, 400 of which were native to Germany. Fuchs gave each plant a German name as well as the traditional Latin. Medicinal uses of aloe, for example, were described by Dioscorides in the 1st century. Fuchs described nearly 100 northern European plants, though, that were unknown to the ancients. This is the most important English herbal prior to the Lynx edition of Hernandez. Gerard directed the gardens of William Cecil, Lord Burleigh, who was Queen Elizabeth's chief executive. In this capacity, Gerard maintained contacts with naturalists around the world who sent him back to England both exotic plants and the soil to grow them in. Turkey corn was cultivated by the Mayans as a staple crop. Although maize originated in Mexico, it became known as turkey corn because it was shipped to Europe through Turkey in the Ottoman Empire. Hernandez later included long and detailed descriptions of corn and the varieties of drinks and meals prepared with it. Gerard includes the earliest printed illustration of the Virginia potato. The history of the world would have been quite different if the potato had never been brought to Europe from the Americas. Gerard grew and described many American plants, including pumpkins, gourds, and squash. It makes me think that reading Gerard is like enjoying a Thanksgiving meal while reading Shakespeare. And there are medicinal plants such as hemp and the wild poppy. And now we come to Hernandez. So Philip II, the King of Spain, commissioned his physician, Francisco Hernandez, to compile Native American natural history and medical knowledge. During the 1570s in central Mexico, Hernandez worked closely with indigenous physicians and indigenous artists. With their help, he described 3,000 Mexican plants. During 3 years of travel, followed by 3 years more working in a Mexican hospital, he studied local medical practice and medicines. Hernandez eventually returned to King Philip, bringing a manuscript with him along with the paintings, seeds, and specimens. After numerous delays, the king passed the manuscript on to his physician and science librarian, Nardo Antonio Recchi. Recchi compiled an abbreviated, edited copy before the original was sadly destroyed in a calamitous fire that consumed much of the King's Science Library. Recchi's manuscript made its way to Naples, to the collection of Ferrante Imperato. While in Naples in 1603, Chasey met with Imperato, viewed his remarkable cabinet of curiosities, and saw Recchi's manuscript of Hernandez. Rumors about Hernandez's work were now proliferating widely, being published in fragments here and there. A full edition was now eagerly awaited and anticipated as a guide to the fountain of youth. Chasey acquired the manuscript and showed it to Galileo. Galileo expressed amazement at the wealth of plant knowledge relayed by Hernandez, entirely unknown to Aristotle and Pliny. As he wrote to a friend in Rome, "In the last few days when I was in the house of His Excellency, the Marquis Chasey, I saw pictures of 500 Indian plants, and I was expected to affirm either that this or that one was a fiction, denying that such plants were to be found in the world. Yet neither I nor anyone else present knew their qualities, virtues, and effects." Publishing a definitive edition with commentary and illustrations comprised the central goal of Chasey and the Academy of the Links. Stoludi eventually published the Links edition in 1651. The title translates thesaurus or treasury. Just imagine what a treasury this many new plants would be. Of the medicinal goods of New Spain, or a description of the plants, animals, and minerals of Mexico. Multiple title pages reflect the chaotic history of revision and editing, or description of the new plants, animals, and minerals of Mexico. The OU copy is unique, containing throughout many pages from an earlier draft, Links hastily printed in 1628. The definitive Links edition incorporated approximately 800 woodcuts. Locating specimens and creating the illustrations proved very challenging and caused many delays. Note the several names. The new plants resisted classification according to traditional European categories and medical qualities. Let's look at a few. Hernandez provided the first written account of vanilla. Nothing like Mexican vanilla. Cinnamon makes an appearance, and pineapple. The heartwood of the guaiacum tree, or Lignum sanctum, was used to cure syphilis. Hernandez described many varieties of chili peppers as a purgative for phlegmatic humors. This plant is described as an extraordinary medicine used a great deal by the natives for the crushed roots, stimulate the appetite, ease bellyache, combat cold, get rid of flatulence, cure an itchy scalp, and kill lice. A handful of crushed leaves dissolved in water resolves swelling. Of this plant, Hernandez wrote, "The gum is clearly hot in the 3rd degree. It binds loose bowels when taken with water in a dose of 1 scruple, and is a good cure for dysentery. It also eases when smeared pains and swellings. The plant has flower-like corn kernels that burst into stars when heated. Is a tall tree with leaves like the citron. The flower seems like the wild rose in shape and has the savor of a Spanish rose. It is lovely in appearance and highly valued for its extremely fragrant flowers and would make a fine adornment to the royal gardens. Applied, the flower eases toothache. Taken, it cures surfeits and chest problems, aids digestion, and it is mixed with chocolate drinks to sweeten the breath." Just imagine the process of assimilating these new medicines. And here's the earliest detailed account and the first illustration of peyote. The peyote plant is hot in the 4th degree. It cures syphilis and mitigates pain, which is caused by chills. It relieves flatulency and removes tumors. If mixed with a little resin, it aids in a remarkable degree in cases of dislocations, fractures, pelvic troubles of women. The seed has some medicinal use. If pulverized or taken in a decoction or used as a poultice on the head or forehead with milk and chili, it is said to cure eye troubles. Formerly, when the priests wanted to commune with their gods and to receive a message from them, they ate this plant to induce a delirium. 1,000 visions and satanic hallucinations appeared to them. In its manner of action, this plant can be compared with the Solanium maniacum of Dioscorides. It grows in warm places in the fields. These are the kinds of descriptions Galileo was asked to comment upon. The Links edition of Hernandez transformed old world natural history and medicine. Its influence appears as late as the botanical works of Erasmus Darwin, grandfather of Charles Darwin, who cited the Hernandez description of manzanilla or chamomile. After Hernandez, European medicine could never be the same. It became necessarily global. So that's medicine in the story of Hernandez. Now let's turn to Galilean innovations in anatomy. But first, Galileo's extensive involvement with the world of medicine is relatively unknown. We'll preface this section with some general remarks about that. Galileo poignantly wrote, "I have been in my bed for 5 weeks, oppressed with weakness and other infirmities from which my age, 74 years, permits me not to hope release. Added to this, O misery, the sight of my right eye, that eye whose labors, dare I say it, have had such glorious results, is forever lost. That of the left, which was and is imperfect, is rendered null by continual weeping." On the most basic level, healthcare affects everyone. Publication of some of Galileo's books, including the famous Dialogo, was held up for years due to an outbreak of plague. Galileo's daughter served the healthcare needs of many, including sisters in her abbey and Galileo himself. Yet Galileo's engagement with the health sciences was extensive. We've already seen this with the thermoscope, the microscope, and Hernandez. But why did physicians study astronomy? This zodiac man diagram portrays a human body surrounded by the 12 signs of the zodiac. Zodiac man diagrams explained the hidden correspondences that were believed to connect the organs of the body, the microcosm, and the influences of the stars and planets, the macrocosm. This zodiac man was published in a book of astronomy and meteorology. It also the first published defense in England of the sun-centered system of Copernicus. Here's another zodiac man. The ability to perform technical astronomical calculations was one characteristic which distinguished university-educated physicians like you from other health practitioners out there in the broader culture. In most universities, astronomy at an advanced level was taught within the medical school. So in this light, it's not surprising that Galileo entered the University of Pisa to study medicine. Like many astronomers, Galileo himself was professionally trained in medicine. Robert Flood was a London physician contemporary with Galileo. In this work, Flood explored correspondences and influences between the macrocosm and the microcosm. According to Flood, the universe itself is a musical instrument, a monochord. The universe can only be understood through music. Likewise, an understanding of medicine depends upon the physician's comprehension of astronomy and music, both of which affect the temperament or tuning of the human body. Does ATSU have a professor of music and astronomy? Let's consider Shakespeare, a contemporary of Galileo. Another reason medical students mastered astronomy was the entire framework of humoral medicine evident in Renaissance literature. According to the ancient medical traditions of Aristotle, Hippocrates, and Galen, the human body, like other bodies beneath the moon, is made of the 4 sublunar elements, earth, water, air, and fire. In the physical world, these 4 elements arise from the four contraries: hot, cold, dry, and wet. In the human body, the contraries give rise to the four humors. Hot and wet make sanguine or blood. Hot and dry make choler, which is yellow bile. Cold and wet make phlegm. Exactly. Cold and dry make melancholy, which is black bile. Health prevails when the four humors are tempered in harmony. The connection with music. Persons are said to possess choleric, melancholic, phlegmatic, or sanguine temperaments, or tunings, when their distinctive balance is tuned toward one or another particular humor. In contrast, Shakespeare described Brutus and Julius Caesar as possessing the elements equally mixed. Now, planetary and stellar influences affect one's physical temperament, so one must take steps not to catch the melancholic influenza of Saturn. Galileo once served as an expert medical witness in court, giving testimony pertaining to melancholia. And let's see. Or there is the choleric influence of Mars, the phlegmatic influence of Moon, and far better, the sanguine influence of Jupiter. That is why Galileo cast horoscopes, such as this one for Cosimo Medici. Galileo cast horoscopes for his family and friends to help them understand and better manage their temperaments. So educated readers believed that a planetary influence or influenza might affect the course of health and disease. University-educated physicians depended upon their ability to prognosticate planetary positions. They used mathematical astronomy to prescribe the appropriate times for therapies, whether bloodlettings or administering medicines or whatever. Are you coming down with the flu? Take 2 pills each morning with the rise of Jupiter. Galileo's second printed work was a defense of his compass, which had been plagiarized by a former student. I mention it here because he inscribed the OU copy to a friend who was a Venetian physician, moved in medical circles in Venice. We have already mentioned Santorio in our earlier discussion of the thermoscope. Santorio was another physician friend in Venice. Santorio is also known for pioneering quantitative studies of metabolism. Santorio designed a weighing chair suspended from an enormous balance to standardize body weight measurements. He scrupulously weighed his dietary urine and feces over a period of several decades and concluded that the greater part of weight one consumes must leave the body through insensible pathways. Quite a science project. This is the first printed edition of the collected works of Hippocrates, collected and edited by Girolamo Mercuriali, a leading physician. Hippocrates was famous for his many aphorisms, including, "Life is short, the craft is long," and "Cure rarely, comfort often, care for always." The title page illustrates the various figures of ancient medicine, including Galen and Hippocrates. The Hippocratic Corpus was written in the 5th century BCE by many authors over more than a century who were associated with a center for health care located on the island of Kos. Mercuriali collected the various Greek texts of the Hippocratic writings and published a critical edition bringing them all together with a parallel Latin translation. A professor of medicine at the University of Padua, Mercuriali was also an acquaintance of Galileo, and he recommended Galileo for a chair of medicine at the prestigious University of Bologna. Galileo did not get the position, but this shows the way he was known in medical circles. So I hope that I've illustrated the extensive involvement of Galileo with the world of medicine. We now turn to Galileo's contributions to anatomy by referring to an unlikely source, a book of literary criticism. Here, Galileo critiqued giant tales by popular writers of the day, such as Tasso. Using his understanding of comparative anatomy and the physics of weight, Galileo refuted the literary description of giants with physically impossible proportions. By the way, Galileo also gave public lectures on Dante, so Earlier we mentioned Galileo's Discorsi, the discourse on two new sciences. The two new sciences concerned tensile strength and motion, and we noted that the science of motion includes the parabolic motion of projectiles and the law of falling bodies. But we passed over the science of tensile strength, which occupies the first part of the book. Galileo articulated what would become a central tenet of comparative anatomy, that homologous structures of animals of different sizes will take on different shapes rather than just appear larger, because the force of weight scales in a nonlinear fashion. When scaling up, one can't just make things larger. In other words, the bones of a giant will not look the same except bigger as those of an ordinary human. They will need to be thicker. Eventually, a large enough giant would simply collapse under his own weight. Thus, giants as described in the Tales of Tasso must be rejected by the principles of the new science of tensile strength. Borelli analyzed the musculoskeletal system in terms of the mechanics of the lever and other simple machines, applying Galileo's physics. Borelli, a practicing mathematician and engineer as well as a physician, studied under a student of Galileo's. He was a member of the Academy of Experiment in Florence. The Museo Galileo in Florence has preserved an anonymous 18th century Borelli-like model of the arm as a third-order lever. And a third-order lever is one in which the effort lies between the fulcrum and the load. So physicians such as Santorio and Borelli, and shall we say A.T. Still, applied to medicine and anatomy the physical principles which occupied Galileo. So that's anatomy. Now let's conclude with a few reflections on innovation by synergy and exaptation. Synergy and exaptation are two forms of interdisciplinary innovation. However, just because something is interdisciplinary doesn't mean it is helpful. The imposition of disciplinary expertise outside its proper domain can be imperialistic or overbearing, as was theology and also physics in Galileo's day. Innovation is not easy to define. Innovation does not mean simply trying something new, nor is innovation at first easy to recognize when it does occur. Innovation often only becomes evident in retrospect. Innovation is not a management technique that can be directed to produce immediate outcomes. Rather, innovation arises from a culture that creates possibilities for synergy and exaptation over time. So to clarify our thinking, let's focus on those two concrete and specific forms of interdisciplinary innovation. When we have spoken of connections between the whole circle of subject areas, we're thinking in the holistic terms of synergy. The whole is more than the sum of the parts because the relationships are essential to each thing. No discipline exists on its own in isolation from other disciplines. Rather, the whole is to some degree present within each part. Now, exaptation originated as a term in evolutionary biology for a trait such as feathers, initially used for heat regulation or for display, but which was later co-opted for flight. So exaptation has come to refer to the productive transfer of traits or ideas between two different domains. The hawk is an admirable example of innovation by exaptation. So here's a longer working definition. When a methodology, technique, skill, concept, question, kind of evidence, data set, or area of expertise arising from one discipline is heuristically applied and then discovered to be organically effective in another. Exaptation is receiving widespread attention these days. We can now attend international conferences such as Exaptation 2018, held on the coast of Italy. Sign me up. We can now read articles about exaptation as a source of creativity, innovation, and diversity in literature such as this Oxford academic journal. But how did a culture arise in Galileo's world capable of creating the possibility of exaptation. Let's consider the renowned Academy of Drawing in Florence. Sirigati, considered earlier, taught at the Academy of Drawing. At that time in Tuscany, many young men enrolled in artisan workshops such as this one. In these artisan workshops, they would study Euclidean geometry in a hands-on way by practicing the techniques of perspective drawing. Later in life, Galileo became an honorary member of the Accademia del Disegno. But as a young man, he studied in the artisan workshop of Buontalenti. Lectures there were also attended by Galileo's friend, the painter Cigoli. This is a painting by Cigoli in Rome of the Virgin standing on the moon. The moon shows craters as it appeared through Galileo's telescope. They had known one another since Buontalenti's workshop. Amazingly, it is reported that Cigoli regarded Galileo as his master in perspective drawing. For their capstone projects, students in these artisan workshops would apply their perspective drawing skills to a project in art, such as a painting or sculpture, and go on to become artists. Or they might make a blueprint and go on to become architects. Or they might create a design for a complex machine and go into engineering, as did Galileo. In artisan workshops, future artists like Cigoli and engineers like Galileo studied geometry side by side. Medicine and physics were also transformed by methods of perspective drawing. And Galileo took the radically innovative step of applying perspective drawing to the telescope. Innovative contributions were made in each of these fields because of their synergistic relation to each other and because of the exaptation or transfer of perspective drawing from geometry. Yet interdisciplinary relations may change in the course of research. In Galileo's day, astrology was successful. An interdisciplinary connection has not endured to the present day. What is the circle of subject areas that come together in your research? And where are your opportunities for exaptation? Perhaps we can imagine some connections. All interesting problems are multidisciplinary. Innovation arises from a specific kind of interdisciplinary culture. Because of the growth of knowledge, no isolated individual today can be a Renaissance polymath. In the modern era, we connect the disciplines in working groups with diverse expertise. Only through collaboration may we discover connections that are obscure in our specialized training. Through exaptation, we may deploy connecting concepts, methodologies, techniques, skills, questions, kinds of evidence, data sets, or areas of expertise in innovative ways. In Galileo's world, innovative applications of mathematics connected art, music, medicine, engineering, and astronomy. Astrology also connected medicine and astronomy. How do we know when an apparent interdisciplinary exaptation will endure? And perhaps most fundamentally, what is our equivalent of the artisan workshop? Thank you. I welcome your comments as well as questions.
Is innovation predictable or unpredictable?
I think we could look back in retrospect, which isn't prediction, but retrospection. We could say the more we understand about Renaissance Tuscany, the more we would be confident in asserting that innovation would take place there. But how and who will turn out to be the innovators in the culture were not obvious until they did it, and often only in retrospect. So what I would say is let's shift our perspective from outcomes and individuals to looking at the culture, which we can know a lot about. How do we produce a culture that produces the kinds of interdisciplinary connections that we saw in Renaissance Tuscany or in other areas? Go back 100 years ago to the founding of ATSU or other areas of creativity that we identify. What can we've been on this time travel journey with our engaging our professional identity zone. So what are the what are the episodes that have formed your particular tradition? What made the steps in that tradition so innovative? How do we translate those cultural conditions to our working ethos today? And that's for you. I'm a historian. I can't do that.
In reading about Einstein, he had, a lot of people say he's the creator of E equals MC squared, but if you really look at the history, there was a number of scientists. So I got to wonder, our interpretation of history is that we have these singular individuals creating something and we put them up on a pedestal, but if we were to really look at the completeness of the history, it was always interdisciplinary, it was always groups of individuals that did this.
Yes, absolutely. So no matter what kind of history of science course you sign up for in our program, that's one of our chief takeaways. Always interdisciplinary, always community-based. When we get beyond the title page and actually look at the community involved, it's also interesting how there are always women present as well in the community. But the example I gave of Galileo in Middle Eastern dress, I think, is a very striking example of this. At the time, astronomers like Galileo and Hevelius were aware that they were standing in a tradition that was far richer than what we in retrospect concede when we say Galileo invented this or he was a founder of modern science. So a professional historian of science is going to emphasize those interdisciplinary and intercultural communications and the communities involved and focus on the culture rather than on a lone genius. Let's focus on culture. That happens to be the central question we face today, too. So it connects with the constructing your own professional identity in terms of the culture that you inculcate.
Question, what characteristics of culture are amenable to innovation moving forward?
Boy, well, moving forward, you just went out of my area of expertise, so that's why I invite comments. All I do is read old dusty books and tell some stories from them. So please, if you can discuss these questions, during lunch, then this talk will be a success. I haven't brought answers. I hope I've stirred your imagination so that you will begin to ask your own questions. Mm-hmm.
Final question.
Oh, I'm sorry. Okay.
Just a comment, maybe. If we look at the culture that you had, it honored, of course, there was this growth and everything, but if we look at each of these individuals, they were all very broadly educated. And I would say then that if we want Dr. Middlemoss's predictor, you can predict that the broadly educated individual will become the innovator. But you also have to have a culture that honors that. And yes. I don't know that ours does.
Yes. Now, just to follow up on that, there's a perfectly good phrase that summarizes this culture, and that's liberal arts. So the Marcianus Capella fellow I mentioned early on, he made that claim about the phases of Venus back in the 4th century when he wrote the book that defined the 7 liberal arts. Mathematics was actually 4 of them were mathematical, and it was working in that liberal arts tradition when astronomy and music were considered as mathematical disciplines related in a sibling way that Kepler found it to make sense to try to make an innovation in astronomy by developing musical theory. Or that Galileo thought that mathematical techniques in drawing were appropriate to physics and astronomy. So Renaissance Tuscany is an example of the liberal arts tradition succeeding, flourishing. What happens if we promote the conversation between disciplines? So we have a great phrase for it. The liberal arts, how do we apply that to our modern educational system at all levels, even the individual working group at an advanced topic. Thank you so much.
Thank you again, Dr. Magruder, for reminding us that we are part of Renaissance Kirksville, not Tuscany.
Yes.
And so just a couple instructions as we head to our next portion of the program. For those that are judges for this afternoon, we will be meeting in the lecture hall next door for our preparation for our lunch. For those who are presenting and for other members of the community that are here, there will be lunch upstairs in the poster session hall. We will be convening the poster presentation portion a little bit before 1 p.m. And half of the presenters will occur within the first hour, and then the second half will occur in the second hour. At the end of that time, we will have our presentation of awards, so please plan on staying through that entire time if you can. So with that, thanks again to all of you participating for establishing this interdisciplinary culture that we call Renaissance Kirksville. Thank you very much.